Okay, it's 10 o'clock Australian Eastern Standard Time, so we'll start this genome annotation using Apollo session. So I'll just get my slides up. Share the screen. Okay, so thanks everyone for joining us today. This, of course, is genome annotation using Apollo web training, and we're very happy to be able to invite or to have Monica Munoz-Torres joining us from California today. So this is an event organised by EMBL-ABR. So I'll just briefly explain what EMBL-ABR is. So this is the EMBL Australia Bioinformatics resource. We have a few aims, and we basically are aiming to increase Australia's capacity to collect, integrate, analyse, exploit, and share large amounts of heterogeneous data sets that are part of modern life sciences research. One of the major things we're interested in is contributing to the development of and providing training in data tools and platforms to enable life sciences across the country to undertake research, and also to enable engagement in international programs that create, deploy and develop best practice approaches to data management, software tools, methods, platforms, services, and also training. So we have 12 nodes around the country. They're shown here on the map. We have, I guess, a number of key areas that we focus on, as I alluded to before. So these are around the data, tools, platform, compute, bystanders, and also training. And training is why we're here today. So we're extremely happy today to be able to offer this genome annotation using Apollo training session with Monica Munoz-Torres. There couldn't be a better person to be delivering this training because Monica is previously the lead of the Apollo project, which is based at Berkeley. Currently, she's working for Phoenix Bioinformatics, a company leading scientific software development. So Monica is joining us from the Bay Area and has graciously offered to do this after hours. I think it's probably coming up to about 5 o'clock there now, or just past. And then at this end, on the other side of the Pacific, we have 9 nodes that are taking part today. So we have a number of sites in Queensland, 3 sites in Brisbane, Townsville and Cairns. We have Lismore and Sydney in New South Wales, 2 sites in Melbourne, 1 in Adelaide and 1 in Hobart. So we're very excited about being able to offer this training through this method. So just a little bit about logistics. Each site here in Australia should have at least 1 previously trained facilitator to help. So all sites will generally be muted throughout the session, and if you want to raise it or ask a question to Monica, you can raise your hand by typing a question into the message, into the Zoom box. So we're very, this is actually a new model we're trialling for training delivery for Australia's bioscience community. So it's, I think, technically potentially challenging. We'll see how this goes today. If there's any facilitators, if there's any particular technical challenges you're facing at your site, please let us know through the chat box, or you can send an email to me directly. Before we start, I just wanted to say a huge thanks to Monica for agreeing to deliver this training. Monica's delivered this training, I think, to many, many people around the world, and we're very, very happy that she's been able to do this for us today. Also want to make huge thanks to Monica and also Nathan, who's on the line from the Apollo project, and also Nick Rhodes at QCIF and QFAB for setting up the training environment. And I also wanted to thank QCIF and Melbourne Bioinformatics for resourcing the training environment. And lastly, also the local facilitators who've done a huge amount of work organising the local training venues. So that's probably all I'm going to say now. And so over to Monica, and the training can start.
Thank you. So we'll start moving things over here, and I'll take over the screen. I'm just going to share my entire desktop. As Jeff said, this may be a little challenging at the beginning as we figure out the logistics. I promised I would put my mic a lot closer, so it's closer now. Hopefully everyone can hear. And in the spirit of being able to get everything set up, I wonder if having the screen like this is better, or if having the full screen is best. Does it make any difference on your end?
I think the full is good.
Okay, brilliant. And Jeff, I will rely on you to help me with the chats, because I will not be able to go through the chat window. So please let me know when it's time, if there's a question, if I need to stop, okay?
Absolutely.
Oh, brilliant. Welcome, everybody. This is incredibly exciting. As Jeff was saying, I've done this training several times over before. I think a total of about somewhere in the vicinity of 1,000 people have been trained the way you are about to be trained, in 50, 60, some presentations over the past 3 years. And I'm hopeful that even though we're not sitting in the same room, the logistics that this amazing team has taken into consideration to make sure that there are facilitators with you will actually make things smooth and just as well as if I were with you in the room. So I think we have approximately 100 people who signed up for this, counting some of us facilitating, of course, from many countries and from every scientific background, and I'm very excited about this. So just take into consideration this as I go through things. There may be things that you're a lot more familiar with and things that you're not so familiar with, and so not with an intent to insult anybody's intelligence, but rather to draw a baseline from which we can all get more information about some of the concepts that I'll touch. As Jeff said, I'm Monica Munoz-Torres. I'm over at Phoenix Bioinformatics, and I formerly worked for Lawrence Berkeley National Laboratory, which is the place where Apollo has been developed for the past 13 years, I believe, almost 15. And so what I would like to do today is to bring you to this concept of what are effective ways to get valuable information from a genome through curation efforts. And if you have heard of the word curation before in a different context, it's probably in the context of a museum. Imagine how everything in a museum has a label that allows you to understand a little bit more about the specimen that is being exhibited. In the same way, think of genomes and genes in that way as we go through this concept of curation, trying to attach metadata to anchored positions in the genome so that we can offer some of this information. Up until now, I've been very focused on structural curation, and I'll tell you more about it in a second. And then as I move into the new workspace in which I am, things will morph a little bit more into functional curation strategies, more the emphasis of Phoenix Bioinformatics. I'm hopeful that over the next couple of hours, you'll be able to get a sense for what the context of the concept of curation is in the context of genome annotation, going from the assembled genome to the automated annotation process to this manual annotation process we're going to do today. And you'll become familiar with the environment for the software, as well as you'll learn about the concepts of identifying homologs of known genes so that you can the homologs of known genes into your genome of interest so that you can go and curate them. And hopefully you'll be able to learn a little bit about corroborating or modifying those predicted genes using the evidence that has been put up for you on the browser. So this is what the evening is going to look like, more or less. We'll talk about genome curation at the beginning, and I'll enter into some detail on predicting and annotating genes. To do that, I'll actually go through a little bio refresher of some concepts that are useful to remember, things like splice sites and what they should look like, what do they look like when they're non-canonical, and the exceptions as well. And then I'll talk specifically about the software. I'll give you a runaround of the land with a little bit of examples on how things happen. I'll walk you through an example that'll give you some insight on how my thought process works when I am working with a gene. I'm in the dark now. Anyways, and after that, we'll do some hands-on practice. So we're actually going to work with some of these exercises. We'll take breaks through the time. I believe that we have a time frame of approximately 3 hours scheduled for this. Correct, Jeff?
That's right.
Yeah. So my introduction will take about 45 minutes to an hour in the beginning. And then we'll do a little bit of logistics to try to figure out how things will work with the exercises and the instances and how you're logging in and all of that. So you don't need to worry right now about logging in on everything. These slides are available online on the GitHub repository for the Apollo project, Genome Architect. But I know that Jeff and the team also made them available to an email to you. So if you don't catch this right here, you'll be able to see them. I sent all of this information ahead of time, and they should be available to you as well if you want to follow along. And with that, then I think we're ready to start. With the meat of this. Are we good?
All good, I think.
Brilliant. So if you think of this is kind of like a quirky little thing that we came up with a few years ago with collaborators about the concept of sequencing a genome. So the whole idea of a genome sequencing project. And we called it a genome train, just because we think that there are all of these different stations that we should go. And, think about the experimental design, think about how things are going to come together into an assembly and how you're going to determine the predicted genes, and then how are you going to curate those genes? How will you assign functions to the genes? And finally, how will you be able to analyze all of your data and bring all of the valuable information out of the genome? So if we define this genome sequencing project as a whole journey through the stations, today we're going to talk about this particular station, which we called the feature curation crossing. So this is what we'll be focusing on. What is it about curation that will facilitate extracting knowledge from the data that we got for the genome? I like to think of our work here as unlocking what's inside of the genome. And there are many things that are done, in the automated world. We have things like understanding the percentage of GC content, as upper here on the right. These graphs show us this is for the honeybee on the left and a little parasitoid wasp in the middle. And so those distributions about GC content throughout the genome, as well as understanding in the far bottom left, the gene composition for your organism and how these unique copy orthologs are represented across evolutionary distance. And so here, for instance, we're simply looking at a tree that shows us what are single copy orthologs present in all species of these particular collection of insects. All the way to arthropods, actually, because we have Daphnia and Pedicellus humanus here as well. And so we can get insight into that composition by understanding the structures and then making global comparisons. And we can also understand a little bit about networks and about molecular or gene family membership for each one of the gene structures that are defined by associating this information to things like the gene ontology. So the terms of the gene ontology. So this image in the middle shows us a little bit of information on some biological processes and how some genes are specifically associated with those biological processes. In general, we unlock the information inside of these genomes to try to understand what makes my organism unique, what makes my organism that organism of interest, and how are they compared to other organisms, what are the similarities to others, and what makes them unique as well. But to get all of that information, then of course I'm here to tell you that you have to have good gene models. And so through the process of sequencing your genome, you generate the gene models, with a few rounds of gene prediction. Whether or not you're training those gene predictors depends on the strategy that you're choosing. And then you annotate those gene models by adding a little bit of information regarding function or expression patterns, experimental data that are available. And we suggest that you manually review both structure and function for all of those genes in order to improve the quality of those genes. And so curation helps you to improve that quality, because you're both trying to extract the best representation of the biology, and you're trying to get rid of the majority of errors that are inherent in these analyses. And then you're also trying to associate the genes that you're looking into with functions by using comparative analysis through the literature, or by comparing sequences, or by comparing your own genes in assessing what their functions can be with experimental data of your own. So the lab that I just left over at Lawrence Berkeley Lab works with those two dimensions by generating the resources of the Gene Ontology Consortium. The cyber infrastructure for the GO actually lives at Berkeley, and also by developing and maintaining Apollo. And so Nathan Dunn, who's in the audience with us today, is actually the technical lead for the Apollo project. And so he's responsible for all the great improvements that the software has seen over the past 3 years. So we can make sure that you get his contact information so you can ask all the questions about all the technical aspects with Apollo. So in this effort for curation is valuable. It'll help you make accurate orthology assessments. It helps you to accurately annotate expanded and contracted gene families, and also it helps you look into what are novelties for the specific gene of interest that you're looking at, or whether there are species-specific isoforms in general, to take advantage of all of this experimental data that's becoming available more and more for all of the genomes. I also like to share this because you will experience this today. Curation is an inherently collaborative process. It's very difficult for a single person to curate an entire genome the way it is needed to be done. And so this represents a problem of scale. But also, it represents an issue of expertise, because we need to be able to find areas of knowledge to which we're not very, where our knowledge is not very deep, right? And so I may be very good at curating genes on the Hox cluster, but it's better for me to get together with someone who works on, say, for instance, odorant receptors if I'm working with an arthropod genome, so that they're able to lend that expertise to us. An example of how this collaboration has helped propel some of the genome projects that have been published in the recent years. I usually talk about this very near and dear project to my heart. I'm one of the members of the steering committee for the I5K, which is a project to try to assemble a community to help in the sequencing of 5,000 arthropod genomes. We have a little ways to go. We've sequenced 60, and then we continue to promote this as a way to help others. And so the I5K actually has a really good collaboration for curation going on. We've trained, as I said, about 1,000 people, and there's an active pool of 400 curators that can be reached out to if you want to collaborate in a genome project that involves an arthropod. This particular project is important because, of course, it tries to tie together all of the information from many and diverse arthropod genomes, thinking in the context of helping understand better things about worldwide agriculture, understanding things about food safety, increasing our knowledge and understanding of certain models in biology, and also at a more fundamental level to walk down every branch of the phylogeny. So one brilliant example that you probably saw in the news a couple of years ago was about the genome of the bedbugs, and how international travel and commerce and has basically brought a resurgence in some areas of the world for bedbugs. And also more and more there's an increased insecticide resistance being reported. And so This Professor Joshua Benoit actually put together a group, the majority of whom were his undergraduates, whom he forced to take this class. And they were actually recruited and trained into this curation process. So they sat down and did a fantastic job curating a few thousand genes with the collaboration of this larger group. But it's important. So everything you see here in red in this image are general characteristics that were very well known for bedbugs, and everything you see in black are actually things that were now identified and knowledge was expanded on because of the process of manual curation that took place. Things like, for instance, understanding that there's an increase in specific genes that are associated with blood digestion, which was speculated but had not been experimentally defined, and several other aspects. All in all, this is timely for biology of human ectoparasites, but also allows to think forward about possible strategies for management for this particular insect. And this opened the door to many of those things. So this is just an example of what you can accomplish by coming together and doing these processes together. So it's not just about arthropods, of course. I'm a little arthropod biased, so the examples that we'll work with today are actually using the honeybee genome, but this is also, Apollo has been used also for several different species of plants, for corals, microorganisms, both eukaryotic and prokaryotic, as well as there's an instance with human data as well. So what I will go over in the next few slides is a little bit about predicting and annotating gene structures so that we can better understand how to refine them. I've seen a few messages come through the chat. Is there anything I need to stop and answer?
Not at the moment.
Brilliant. So let's start with the concepts of gene prediction and gene annotation. So we're basically trying to identify and annotate genomic elements, right? As the machine, as we lay out our genome, we try to identify, primarily focused on protein coding genes, but also identifying certain RNAs, regulatory motifs, repetitive elements. And so this process of gene prediction and gene annotation happens in two steps. There's a computation phase and there's an annotation phase. And I'm going to explain a little bit more about this computation and annotation phase. All of this actually comes from a very cool paper from 2012. Mark Yandel and Daniel Enzi wrote about this. So I put the reference there. You guys can revisit this in your own time. It's a pretty cool paper. So the computational phase actually has two steps as well. So there's this part of experimental data that is being aligned to the genome. So you're using proteins and RNA sequencing reads, and you align all of these to the genome. And as well, with that information, gene predictions are generated, right? And so you can have an ab initio process, which is solely based on nucleotide composition, and that's how gene predictions can be generated. Or you can do gene prediction using experimental evidence and support of these predictions, which is going to allow you to identify domains and motifs and things like that. So for the ab initio part, so when you hear us talking about ab initio prediction, basically, as I said, you're talking about everything is based solely on the DNA composition without taking into account other information. And it deals strictly with genomic sequences using statistical approaches to be able to search coding regions and to be able to identify. So the image to your right basically says, okay, so I have this genomic sequence, and the gene model can be defined even if there's all of these components of initial exon, internal exon, final exon, and UTRs. Sorry, that's a typo on the right from the image. I forgot to overwrite it. It's a 3' UTR on the right. The problem is that with ab initio predictions, it's a little difficult to identify the possibility of alternatively spliced forms of the same gene, right? So the output down here at the bottom shows us there's, perhaps there's one gene predicted at the beginning, and then one gene in the middle, and one gene at the end. But that internal exon up here at the top, which would have given a differentially spliced form of the gene, is not taken into consideration because the predicting algorithm is simply going by the DNA composition and nothing else, not taking any other cues. So an improvement on that is the evidence-based form of gene prediction. So you're actually finding genes using sequence similarity searches. You're using that against publicly available databases, or using experimental data sets that you can do in order to inform the prediction of these genes. And so that's what, for instance, the FGenSage++ pipeline does. As part of the computation phase, then the result is we get what is the single most likely coding sequence, although no UTRs are annotated, and usually we don't get any isoforms annotated. Then the annotation phase actually brings all of the experimental evidence added into this process, and we allow the prediction tool to synthesize all of this information into structural gene annotations. As a result, at the bottom, you can get gene models that you can see generally include untranslated regions, and also would show you isoforms. So here you see this is an isoform where the start codon's over here at the end of the second exon, and then this form, this spliced form, actually has the beginning in third exon. And then you have evidence in order to support these hypotheses. After you've predicted and annotated these genes, then gene models may be organized into sets. And that's why you're going to hear me talking about the official gene set. So that's after several processes of gene prediction have taken place, either a combiner or a pipeline has been used in order to synthesize all of this information into gene sets. So here I talk about a combiner, for instance. You automatically integrate these genes into sets using something like evidence modeler that you've probably heard talk about, or Glean, or Mark Yandel, the author of the paper, his lab produced this piece of software called Maker, which is basically a pipeline that allows you to bring all of this information and generate a consensus gene set out of all of the gene predictors that are included. So you can get good results using just the ab initio predictors. You can capture things that are specific to that species because you're not looking for external evidence or cues in order to define those genes. So you're actually able to define things like highly divergent genes. But sometimes this information is not enough to establish orthology for those gene models. And of course, false positive predictions are always possible. And on the reference guided one, on the other hand, you can reliably use orthologs from species that have been annotated, but you can miss that specificity of genes that only belong to this one species that you get with the ab initio prediction, as well as other downfalls. So the suggestion is to always do a combination of the two. Do a hybrid reference guided and ab initio gene prediction system so that you can you make use of the transcriptomic data to be able to confirm those predictions, and then so that you're able to review synteny to verify orthologous assignments across different species. So this is all about defining the structure of the gene prediction annotation. But then the step forward after that is to define gene functions or to make functional assignments. So functional annotation, I basically define it as attaching metadata to those structural annotations for the purpose of assigning a function. And so this can be used most typically is done using terms from the gene ontology. But you need to understand that assignments are not necessarily supported by your own data, so you can do this by sequence similarity, but those sequence similarity approaches have to take into consideration evolutionary distance, evolutionary relatedness, and phylogenetic information in general. So not just the score value with BLAST. Oh yeah, these look good, they should be the same. Remember that if you do that, you're probably getting into this problem of transitivity where you're, a bad annotation in one of those genes, and if your gene looks like that one, then you're actually moving the annotation across species with subpar information. So not only the sequence similarity scores, but also informing and validating this information with phylogenetics will be the best way to make this approach. What does that look like? Well, making comparisons using software that actually builds HMMs, hidden Markov models, or that actually looks at domains that have been defined by an algorithm like interproscan, right? So that would actually allow you to make searches against the database that has been curated and has functional assignments, as I mentioned before, from things like the gene ontology. Why do I mention the Genome Ontology? Of course, because I used to work for them, but also because this is actually a very good collection of classes that give you information about molecular functions, biological processes, and cellular locations that are associated with a gene. And so in terms of molecular function, if you have heard this, then forgive me for the very quick explanation about what the Genome Ontology entails. So we define a molecular function as an elemental activity, right? And so in this case, this funky looking receptor is called, it's actually a kinase. And it's an insulin receptor, sorry, and it has a protein kinase activity and also insulin receptor activity. A biological process is then a commonly organized, recognized series of events that are organized in a specific manner, say cell division. This is a photograph of the end of telophase. And the cellular component is the localization of a gene product. And so this is a beautiful image of a mitochondria. The cellular component could be the mitochondria itself, the matrix, or the inner membrane. So all of that information allow us to enhance the knowledge that we have about each one of our gene structures. And usually I like to share that this is a collaborative process. This doesn't happen in the ether and magically comes up. The ontology is actually maintained by a team of about 150 contributors from many institutions around the world. And so make sure you can make use of this information to try to make functional assignments. And that's the end of what I will say today regarding functional assignments. If you have any more questions, you can feel free to shoot me an email and I'll do my best to answer or redirect as necessary. Then we get to talk about Apollo. 25 minutes into it, we're going to start visiting the reason why we got together today. So Apollo is a piece of software that allows us to curate gene structures, but not just by yourself. It allows you to do this in a collaborative manner so that you can overcome those challenges of scale and expertise. In general, here's how I view this process of curation when you're using Apollo. Before we look at the window, think about these 6 steps. First, you select a region of interest that you have identified as the possible target. We'll do that as the first step in all of our exercises. And then we look at the evidence track. So all of that information that has been aligned against the genome in support of those gene hypotheses, because that's truly what they are. And you select the appropriate evidence track that will allow you to annotate gene models in the region that you're interested in looking. Once you see a gene model, or any recognized feature, genomic element in an evidence track, see if you can determine what would be a good starting point for your gene of interest. This basically means, has there been a prediction of a gene model in this particular region that I just found sequence similarity to? And if so, that's your starting point. And if necessary, you will adjust that gene model depending on the information that you have. And once you adjust the gene model, you check the finalized edited gene model for integrity and accuracy. And I'll repeat several times over what this is, but it basically entails looking whether or not you found the things that are usually found in a gene: a start, a stop, canonical splice sites, UTRs. Are there any gaps? And you want to compare that to homologs available in publicly distributed databases. And finally, you'll add all of the information regarding what you just did to the model. So I know this sounds very ethereal right now because you haven't even looked at the Apollo window, but I'd like for you to get into the mindset of those steps that are going to take place in order to annotate a gene. Find it, see if there's any evidence of its existence from the experimental data, of its definition. See if you need to change anything, because if you don't, then you just click away and you're done. Make sure that you compare what you have been working on with everything available in public databases, and make sure that you keep track of all the changes that you've made. And that's the basic concept of curating a gene model. So for us to get into this business, I'm going to give you a tiny refresher on a few things that I consider very important in terms of definitions for, little biochemical tricks that we might have put in the back of our minds. So first, what is a gene? And I know this is a discussion that it's 150 years old. So by no stretch of my imagination, we're going to define exactly what it is and make everybody happy. So let's give it a try by talking about what we understand as a gene when we're doing structural curation. So we think of the gene as this union of genomic sequences that are encoding a coherent set of potentially overlapping functional products. Why potentially overlapping? That might be the flag that you identify right away. So if we look at this image at the very top, we have this drawing telling us this is what the DNA would look like just laying down on a stretched piece of chromosome. And we have identified these 5 elements that are flagged as A, B, C, D, and E. If we were to release the transcription machinery into this piece of DNA and allow it to walk through it, and the transcription machinery would identify these many primary transcripts. The first one would have elements A, B, and C, as well as some UTRs over there identified. The second one, we have elements D and E, as well as some UTRs identified. But there would also be an additional primary transcript that has elements Y and X, potentially overlapping products. Let's see if they're functional. So once these transcripts, these primary transcripts are spliced, here's what we get. We actually get several spliced forms of that protein on the left. We have a spliced transcript that includes exons A and B, a spliced transcript that includes exons A and C, and another overlapping one with B and C. As well, another one that shares another pair that shares a UTR on the 5' end, but the actual coding sequence is not shared. They're not overlapped, right? So that's what we see on D and E. Again, that spliced transcript at the very bottom is just labeled as X and Y. When we look at these from the standpoint of functional products, whether these are functional proteins or not, we see that there are 3 functional proteins on that group on the left. They all conserve some, they all overlap to some degree, right? So A and B, or A and C, and B and C all have some overlapping. So we consider that entire group over here in yellow, A, B, and C, as one gene, differentially spliced, but all components of the same gene. Over here on the right, D and E share an untranslated region, but there is no sharing of the coding sequence. Those are going to be 2 different genes, even though they share untranslated regions. And then this last one over here, this non-coding RNA that we find at the very bottom, is considered another gene. So those are the things that we're going to think about when we're looking at the genes. And those are the paradigms that we'll contemplate when trying to define the structure of a gene. It won't get this complicated with the exercises that I'm giving you, but it's good to know because you might come across this information later on when you're working with your very complicated genomes. If you're working with plants, it gets very interesting. So this is what the mRNA looks like. I know you know this. This is your best friend, so always remember that you have exons and introns, and that the exons include both coding sequences and untranslated regions. So it's possible that there's an untranslated region in your exon. Maybe you won't see it identified, but if you do, remember that that is also part of your exon, especially when you're looking at the sequences for your gene models in typed form. So not visually looking at a diagram on a browser, but you're looking at the sequence laid out in coordinates on a tabulated format, text format, also known as the not very standard GFF3. So in addition to understanding that, remember what a reading frame is. Bring that back to the front of your mind, remembering that in eukaryotes, there's only one reading frame per section of DNA that's biologically relevant, and it can be transcribed into RNA, and it can be translated into a protein, and that an opening reading frame constitutes a start signal, a coding sequence divisible by 3, and a stop. Always. If all those 3 things are not included, then it's not an entire open reading frame, it's just a fragment of it. And also remember what splice sites look like in the majority of eukaryotes. Again, I'm very arthropod biased here, so I'm talking primarily about the eukaryotes that we're going to annotate. Things may be a little different if you're working with operands. But in the case of the eukaryotes, remember that splicing signals are going to be, if I am an intron, then in my right-hand side, I'm going to have a GT signal, and in my left-hand side, towards the 3' end of, if I'm forward transcribed in the forward strand, then on the 3' end, I'm going to have an AG. You will find this in the exercises, actually, so I'll bring it back when I'm talking to you through the exercise. But there are some less common splice donors, such as GC, which is very commonly found on arthropods, not other eukaryotes, but it is in arthropods. So from the image that you have in front of you, on the left you have the end of an exon, and it's labeled as 5', so that's an exon, and that's the end of an exon, the beginning of the intron, so this is the 5' end of the intron. And there's going to be a GT signal at the beginning of the intron. If this slash were simply the continuation of the intron at the end of the intron, the 3' end, we would identify an AG signal. So those are usually the signals that the transcription machinery picks up on in order to generate the splice forms, the translation machinery. And so this gives, of course, the machinery the ability to produce more than one protein from the same genic region by creating alternatively spliced isoforms. And those can be identified. Sometimes you can see the transcriptomic data supporting changes like that. And it'll be a lot easier to visualize once we're playing with the browser. Then remember that introns can interrupt the reading frame in 3 different spaces, right? It can be right in the middle of 2 consecutive codons, or right in the middle of a codon between its first and second nucleotide, or right in the middle of a codon between the second and the third nucleotide. So any of those positions for interruption are possible, and how that interruption happens will change the coding sequence, the frame of the coding sequence from one exon to the next. So remember, not the entire protein does not need to be coded in the same frame. It can switch from exon to exon, and it can switch from isoform to isoform as well. There are center obstacles to transcription and translation that we have to put back into our mind, and this is the last of the bio refresher. So there are premature stop codons in the message sometimes, and this happened because of a process called nonsense mediated decay, and this actually checks and corrects these premature stop codons to avoid incomplete splicing or to prematurely stop the protein. You can identify those, and there are also natural insertions and deletions that cause frame shifts when the indel is not divisible by 3. So a peptide can be very long if this happened, or it can be a lot shorter depending on where the stop signal is located. And you can correct for these things as well. So if you have, for instance, if you happen to be working with a selenocysteine containing protein, then you will have to annotate a stop codon earlier than normal and change its meaning from stop to a uracil message. But we'll get to that in a little bit. So that's it. That was my bio refresher. So I hope the last 10 minutes have been useful to bring those concepts back to the front of your mind. So now let me show you what that window looks like. This, if you open a browser, and you probably have already done this with your computers, if you've received the credentials, you've probably seen this. So if you open a browser, this is what the window is going to look like for Apollo. On the left, you have at the very top, user-created annotations, and then you also have over here this dark part with the very colorful reads and gene models is where you have all of the evidence tracks. So you have all of the information that supports these gene models, or is information that has been aligned to the genome. As well, each of these tracks is listed over here on the right-hand side, and I'll go into more detail for that little box on the right, which we call the annotator panel. With these 3 arrows, I'm just simply signifying that you can actually enter transcriptomic data in several different forms. In this case, we're looking at stage and cell-specific data that it's different. These are 2 different tissues, or this is for forager, bees in this case. And then over here, we have an actual XY plot of the expression patterns for nurse RNA-seq reads in this particular portion of the genome. Apollo actually allows you to bring up your own information and to put it up into the browser temporarily for the purpose of comparing what your own experimental data shows in that particular portion of the genome. So you can do that as well. You may have noticed that these genes are very colorful. The exons are colored in 3 different colors. It has pink, green, and purple or violet, I guess. And so this is done because you can actually change the color of each exon by the reading frame. By doing this, you actually facilitate the process of changing and editing your gene. This will become clearer in a little bit when you start working. We're going to use a tool to query the genome. So in order to interrogate the genome and find out where our sequences of interest are, we can use something called BLAST. Sounds like BLAST, not quite. And we use BLAST instead of BLAST. It's a similar piece of software, also aligns sequences, but it sacrifices a little bit of the specificity in favor of the speed. So we need to have basically we need to have sequences that are more similar to what is expected to be found. It doesn't do very well with very long strings of not very closely related organisms. Over here at the top, you can see it in blue in the background underneath these browser icons, but I have amplified it over here so that you can see these little tools for navigation. These become very important. You're going to use them a lot. These little arrows allow you, of course, to navigate left and right. And these magnifying glasses, of course, allow you to get in closer or further out from the region that you're inspecting. These 2 boxes allow you to navigate to different parts of the genome. If you know the scaffold that you're looking for, you can enter it here. If the gene model that you're looking for has been already listed in your system, so we do that by indexing, then you can just type the name of that gene and you should be able to locate it from there. And then there's the annotator panel over here to the right. I'll have a lot more to say about that in a little while. So there are a lot of very intuitive gestures that you can do, mostly, like the way you interact with other software on a regular computer. You right click to reveal a few menus like this. And so you can do things like that over here on the evidence panel. And if you right click in the evidence panel, you have the ability to generate new genes as such. Or you can change the annotation type for an annotation that it's already there, or you can work with all of this functionality, which I will be explaining in a little bit. You can also export all of the information. All of those annotations that you have been working on can be exported in bulk, and you can do this selectively. It can also be done programmatically, so your admins can work on this, but you can get all of the sequences, both the FASTA sequences as well as the GFF3 coordinates for every single one of the gene models that you've been working on, so that you can save your work in your own computer if you wish. Now, something that is very, very important that I hope that you guys can make use of is that we are able to facilitate this and allow you to collaborate in real time. So everything, so that, let's say, for instance, that over here on the left is Moni working on the computer, and over here on the right, Nathan's working on the computer. Any genes that Nathan is editing here on the right, if I am looking at the exact same region, I can see, because I can see all the edits, because all of these edits are happening, are being updated to the server in real time. And so they're broadcasted to every client that has them open, which will become important for your exercises, because we've done our best to make sure that you don't bump into each other while you're doing the exercises, but it may just be the case that you notice that you get to a region and somebody else has already started working on a gene there. So that's because you're working on real time with somebody else, and everything is open so that everybody else can see the work that you're doing. This is to foster a spirit of collaboration so that the genes can be improved by you and your peers. And this is what the inner workings of Apollo look like in the back. This is what the architecture looks like. Apollo, basically, all you need to get from this is that Apollo has 3 main components. It has a client side, This is the UI that you interact with in order to be able to make your edits. And on the server side, you have a database that allows you to store and query all of the information for all of the annotations that you're doing, as well as an editing engine that takes care of understanding every single edit, every single function that you're asking Apollo to do in every edit to the gene models, as well as understanding how to respond to every function that you're conducting. So it can pick up that command and it can also respond to it. And if you have more questions about the architecture that are specific to how your project would work, then I am sure that Nathan will be delighted to walk you through that process. As I was mentioning before, over here on the right, there's this box. And this box has basically several different tabs. You will only see 3 of them, and those are the ones that I'm going to explain. The annotations, the tracks, and the reference sequence. The rest of them are boxes that your administrators, or you as an administrator, can work with. So let's start with the annotations tab on the very right. This tab actually lists all of the annotations that have been edited. So it doesn't list those that are aligned to the genome just from the official gene sets or whichever tracks you've added. It only lists those that you are conducting. And when you see an annotation listed, you're going to see it in this little blue writing. I hope that you can see my cursor moving. I'm not sure if you can see this, but I'm down at the bottom left corner of the leftmost figure where it says spell one with a little arrow that says gene. And then underneath it, if you open it or double click on it, you're going to get an mRNA. And this information of your mRNA will have as well, some statistics over here on the right in this part of the table. By clicking on this little arrow, you can navigate to that particular annotation, or you can click on the annotation or find its name from the annotation name search box and then use this enormous box that says go to annotation. So those are 2 ways to navigate to an annotation if you're going away and coming back to your window. From the mRNA box, there's additional information that you can enter or change about a specific mRNA. And that's what I am illustrating over here in this box with the number 1. So these annotation details include information on the name, as well as location, who's the person that has been working on this gene. And over here, the box labeled with number 2 allows you to look at exon boundaries. This is if you were trying to just do a 1-by-1 base pair changes to the boundaries. This becomes more relevant when you're working on your 310th gene model, and you've become a little bit more familiar with how to change those boundaries. But it's good for you to know what those boxes do just in case you come across those. Also in the annotations tab, we have a little bit of information on how to navigate to annotation. I told you that. You can also use the boxes to search those annotations. And as you see here on the drop-down menu on the right, you can look at the annotations by type. If you're looking for a pseudogene or repeat region, you can actually narrow down your searches in that way. You can also display the tracks or remove them from the main window. So you saw that main window on the left had all of those different tracks. You can remove them all. And actually, when you first look at Apollo, it's like you're not going to see any tracks. So it's not that it went wrong. It's simply that they haven't been displayed. So the little green arrow on the left shows us that you can check or uncheck a track to display it. And it also shows over here on the upper left this little line, this little icon to show you that there's another way to look at the tracks, to just basically bring back the list of tracks. If you have used JBrowse before, so Apollo is built on top of a very popular genome browser called JBrowse, and if you've used it before, you may be missing that look and feel of the old style JBrowse track selectors. So for those of you who will be missing it, you can actually display the JBrowse track selector in the same way. We agreed to think that it's a lot more useful to use it on the annotator box, but sometimes it's just a lot easier to look into things that are more familiar to us. So that's why we still make it available. And then the other tab that you can see is the reference sequence tab. So this is basically, we call it a reference sequence because it's the assembled genome that is basically underlying this entire operation, right? So every time you're looking at a scaffold or a window with a scaffold from your genome, there's an underlying sequence. And so that's a fragment of the assembly of your genome of interest. And that's what we're calling here the reference sequence. Because we're working with honeybee, you're going to notice that there's a peculiar naming convention for these fragments of the assembly. You're probably more familiar with the word scaffolds or contings or chromosomes, but these are actually groups. And the reason why they're called groups is because this is the honeybee, and the honeybee has 16 linkage groups and has a lot of molecular markers that have been anchored to the genome. And so the scaffold is actually counted by how many subgroups of each one of the linkage groups exist. So you'll have 1 through 16, and then 16.1, 2, 3, 4, and et cetera, et cetera. So that's why they're called groups instead of scaffolds or chromosomes. It may sound arbitrary, but there's actually a little meaning behind it. So you'll see when we are looking through the exercises, I will say something like, look at group 16.4 or something like that. So when you come across that information, that's what that is about. Okay, we're coming to the end of this tour de force on becoming familiar with what Apollo looks like. And so the last functionality of those windows before you actually go and play with it yourself is that you have the ability to share the location that you're working in. So at the very top, this little arrow shows us a link to links brought together to show you that you can either share a public URL or a logged in URL. That is, if your genome has been made available for visualization in a public manner, not the annotations, but just the browser, then you can share that. If your data are only visible to a restricted group of users, then you should share the logged in URL. That means they have to enter their name and password in order to be able to see it. So say that you're working with your lab mate today, and they're working on the computer next to you, and you want to show them, come and look at the exact same place where I am, but I want you to see it in your computer so that we can both work on it. Then you can just click on that link, pick up the link, and send them by email. And then now they can share, they can find you exactly where you are located. You can also use this drop-down menu at the top to switch organisms. You won't be able to see them because we've made the sandboxing restrictive of the organisms that you're able to see. But I will be able to see everybody's work, so you can only see yours. But if you were able to see others, then they would be listed on this drop-down menu. You can hide or show the annotator panel, so that box on the right can be hidden by using this little caret type thing. And you can also, of course, leave a session by simply using this arrow, either by going to your name and logging out, or using the little arrow to get you out of there. All right, well, as I said, these slides are available in these links or in the emails that you guys received. And I'd like for you to try to play a little bit with the instance for the first time. So I have a filler slide here that has some information on how to enter, but pay no attention to these because you guys have your own entry credentials. What I will explain is, if you look at this thing that a couple of rows that I have highlighted, then you're going to see that you have a number. There's a number for the person. That number means nothing other than, usually I go around the room and I go, all right, let's count 1 through 60. 1, 2, 3, 4, right? So forget the numbers of no consequence here. You have a fake email that you'll need to use as your username. And then you have a password. Look how difficult that is. User one. So that will be the password that you use to enter once you see the first window of Apollo. So do we have, before we go there, I just want to say thanks to Nathan, of course, to Chris Mungle, to Susanna Lewis over at the Bebop team. All of the projects, all of this work that I'm talking about has been done at Berkeley Labs over the past few many, many years. And it's a fun group that I miss dearly, and I'm very happy to give credit for this and many other projects that they're working on. So check them out, berkeley.org, if you want to learn more about things like the Monarch Initiative, the Plantium project, the Inca project, INCATS translator, and a whole other collection of projects that they're working with. But I digress. Let's look into the files and let's look into everybody's connections. I see that the chat is open and active, and let's take a moment to make sure everybody is able to see an Apollo window.
Okay, so I've just put in some shortened URLs for all of the accounts, also links to the exercises and sequences. So how are all of the nodes going, getting people on? Any questions, issues with the environments, or? All good in Tasmania, excellent. Maybe each node, if you could just, all good in Townsville, awesome. Good in Sydney. SCU, Adelaide. Oh, everything's working. The internet gods are being kind to us.
They're liking us today, that's good. Brilliant. I'm going to log out.
It looks pretty good all round, actually.
Okay. Oopsie daisy. We spoke too soon. Okay. I was just going to say this, and here is a very appropriate moment to say this. Everybody can still hear me, right?
Yes,
so,
good.
We are, of course, working with the gods of the internet, and that means that some things will be smoothly and some will not. I, for example, just lost access to the Apollo instance that I was using. Sometimes it happens that we do overload the server and reaching out becomes a little bit more challenging. It could also be my internet. We've tried to make sure that the instances are organized so that we've separated people by all of the people by to try not to overload any of the machines. But we're coming across that little thing. So I'm gonna see if I can find, let's stop this for a sec, and I'm gonna find my credentials for the other one that Jeff had sent me.
Yeah, I'll just send you that email. I'll pop it into the chat. You're the first person on this.
Thank you.
That link there.
Brilliant.
And I had just logged in as you, and it was working for me, so it should be fine. Week 5 is not reachable. I'll have a look at that, Sonica.
Okay, I'm in. So this process, the first time that you are loading the genome, takes a second. So you saw that I just logged in, but it took a few seconds. Once you log in, for now, here's all I would like for you to do. Look at the window. There's nothing in here to begin with, right? As I told you, And for instance, in my screen, I have no tracks. And I cannot see any gene model. So the first thing I would do is to go to the tracks list, right? And I hope Nathan's looking at this. And you can check on one of these. So for instance, I'm going to check on the, let's do Augustus said 12. So you can see the set just came up. And now I have genes, right? But they're all in the evidence track. Then I'm going to check on the official gene set, version 3.2, because that's the one I want. And then you can move things around like this. For instance, you just move the official gene set to the top. There you go. And now the Augustus set is down below, or you don't want to see that Augustus set anymore. You can click on it. You can rubber band to zoom like this. See, I just went to that gray bar and I click and I'm rubber banding to zoom. So now I can see gene models a little closer. Or you can use the magnifying glasses, little step by little step, or you can use the big step of the magnifying glass. Or you can just double click to zoom in. So you can do all of these operations, and I'd like for you to play around with it. Here's how you select a gene, for instance. You can either select one exon. I'm going to zoom in a little bit so that you can see this a little better. I'm going to hide this box to the right because I don't want to see that right now. So if you just want to check, say that you want to look at this gene model because it's of interest, you can select one exon, double click on it to select the entire protein. Or you can click on the intron, and it also selects the entire protein. You can create a new gene model by selecting the entire gene model, right-clicking and saying, create a new annotation, and it gives you the possibility of creating any of 9 types of genomic elements. Here's how a tRNA looks like if that's what you're creating in that region. Or you can simply select the entire gene model, pick it up, and see how it becomes a ghost? And then you can drop it off in that user-created annotations area, and now you have a gene model. To delete, okay. Oh, this is just me. I'm going to go over this, Claudia. I'm going to go over all of this in much detail. I'm just telling you things that you can do as everybody comes up to speed. So don't worry about it. I won't leave you behind. This is just go play. If you want to, you can see things like, oh, I can color things by the CDS frame. Oh, I don't have to do that. Oh, I can only show the plus strand, or I can only show the minus strand. So just play around. You're not going to break anything. If you create a gene model, delete it. It's going to tell you what you can delete. So just delete it. So while I'm playing with these features here, you can be set up. I'm going to go through all of these steps in detail on the other presentation in a few moments. And then you have a slide deck that will have every single one of those functions also detailed. And so don't worry about it. You won't stay behind. You have this little, you see that I clicked again on the side panel to display the box so that we can look at all of the different tracks that we have. There's a lot of data available for the honeybee genome. This is Apis mellifera, the European honeybee, and we have data from 454 contigs that were built. We have RNA-seq, we have NCBI predicted genes, we have several different versions of the same, of the gene set, the original version and a few improvements. And we also have aligned data from closely related organisms. So you're gonna see these tracks, for instance, called ACEP, C-Flow, H-Cell. If you work on arthropods, and if you work with Hymenoptera, then you're probably beginning to recognize that these are the names of other ants, wasps,
There's, of course, Drosophila, and a few other. I think there's also Thermopsis nevadensis, what's that, the termite. So all of these, look at the RNA-seq reads. If I click on it, then Apollo starts thinking. It may show me the details of the reads, or it may show me this announcement. Too much data to show. That's not an error. You can just zoom in closely. And then the closer you get, then the reads begin to appear. And my computer is going bananas. My tiny little MacBook Pro is going, too much on this processor. Okay. So I'd like for somebody, if anyone is working on the, I guess this is the Queensland, this is one of the Queensland instances, right? And NSW as well. And if you're working on server 1, so that would be, yeah, QLD1. And if you happen to be working on Honey 5, let's see if there's anyone with that configuration. Honey 5 on server 1. Yeah, so Tania Duarte. It's probably in the same configuration that I am right now. How much is the browser performance determined by the server power versus the computer running the browser? So Apollo has a component on the client and has a component on the server. The performance is going to be affected by both, not equally, but the issue is that when you're trying to show things that are heavy on the client, for instance, RNA-seq reads, it may just be a little too much. Sometimes if you have like a lot of data that are being downloaded and you're trying to move very fast across those amounts of data on BAM reads, then the browser will crap out, and it depends on more the machine that is running the instance than on the server. However, if there are 30 people attacking the same server at the same time in the same region with the same reads, then the server is going to be the one that's not going to be very happy about it. So it depends on what you're doing, and it depends on the type of request that you're sending. Does that answer your question, SBI? Brilliant. So you can see, I'm making some time just to make sure that, oh, okay. You've avoided VIC5 and we're all good.
I was just going to say that there's an issue with one of the VMs, so VIC5. So what I might do is move people that were on VIC5 onto one of the other VMs. So I'll do that whilst we're speaking, because I think there's a bit of capacity on the Queensland one.
Okay. Sorry to hear that. I know. It's the curse of the live demo, something like that. And it's minor compared to the volume, so I'm happy that that's all. So if you're looking at these reads that I have displayed in my computer, you can see that there are some blue reads and some red lines aligned to the genome. I'm going to get rid of. Oh, Jeff, they're saying not to worry about it. So you can see here, this used to be a little bit easier to see, but I'm going to try to point it out at you. So let's go to an exon. Here we go. If you look at this gene model that is predicted up here at the top, you're going to notice, so these very colorful boxes are the exons, and these thinner lines are the introns of this gene model that has been predicted. All of these lines down here are RNA-seq reads aligned using a BAM file. Basically, you've aligned this to the genome, and now you're using a DSF3 file to locate those coordinates into to the genome. So you're going to notice there are some some portion of the read is darker and some portion of the read is lighter. I'm going to try to get closer so that you can see that usually that lighter portion of the read coincides with the intron. You see that? Maybe you're able to look at that. So darker portions of the reads, or darker reads by themselves over here, typically under an exon. Lighter portions of the read, or lighter, typically corresponding with the intron. The reason for that, as you may have already figured out, is because these are RNA-seq reads, right? So these are actually transcribed regions, which means there wasn't any intron to begin with when these reads were produced. When they are aligned to the genome, well, the genome does have genomic sequence, which includes the introns. And so in order to tie the two portions of that read that belong to these two spliced, the spliced form of the protein, you can link them. We've linked them using this little string. And this little string, it's lighter in color to identify, to basically denote the fact that this is a read that starts here and ends here. And this is just a gap in the middle, but this is still the continuation of the read. Okay, that's important. And it'll become important in some of the exercises because looking at how this alternates will help you identify the possibility of alternatively spliced forms of the same gene. Because sometimes you'll see that an exon has absolutely no reads in support of its existence, except for a chunk of those reads, right? So there's a very big chunk that has no transcription evidence of that exon, and there's a very large chunk that shows, yes, transcription evidence of that exon. And that's most likely because there's a spliced form of the protein that contains the exon and another one that doesn't. And you've recovered transcriptomic data from both. Okay, so that's important to know. Do we think that everybody has had at least a minute or two to play around with the browser, to open a window, to log in? They were able to see what's going on?
So I'm still working on the VIC5 issue. So please just look over someone's shoulder if you were on VIC number 5, and I will get back to these guys as soon as possible.
No worries, no worries. So we can zoom back out, and you can see that the reads disappeared. Here's another trick for you to learn. So say that, let's just bring this one exon up so that I can do it. Say that you want to look at the sequence a little bit up close. So you can right-click and say zoom to base level. So as you see, Apollo has now zoomed all the way to display the underlying reference sequence. And as you can see, I'm going to remove all of this. To just show you the sequences. You see that? I'm going to remove. This is just the reference. This is the actual assembled genome. Forward strand above, reverse strand down below. As you saw, if I right click, I can actually make the protein disappear, the protein translations. Without the colors, these are protein translations in the forward strand, all above the aligned, the assembly piece. And these are all translations in the reverse strand, all below the assembled piece, right? And when I tell it to color by CDS frame, then it's actually showing me every single translation frame for all of the sequence, starting from the 5' to the 3', or the 5' to the 3', from the forward strand or the reverse strand. So you can see all of those. And so naturally what I'm looking at here, if I click on that exon, then I can see, Oh, here's another trick. We're going to get rid of this label. So I can see the exon. And you may be able to see this when you're looking at the window itself, you'll see it better. This is an arrow that shows me the direction of transcription. So here I know that it's being transcribed in the reverse strand. And if I click on the actual exon, it also shows the sequences. And notice that every time I touch the gene model, a little balloon, a pop-up comes up with information on what kind of annotation it is, who's the owner, when was it last modified. So that's information on how to zoom in, and you can also zoom back out. If you don't conduct any zooming operations while you're zoomed all the way to best pair, then you can zoom back out. Otherwise, you can just zoom back out with the big magnifying glasses. And I can just delete this because that is not an actual gene model. And if you saw me doing this, I remove the track label. See how it shows up again? User create annotations, official gene set, and there's RNA-seq reads, but you can get rid of it just to give yourself a little more real estate. You can also tell Apollo to show or hide either direction of transcription, reverse or plus or minus. So you can say, I actually just want to see the plus one, the minus one, the reverse strand. So just get rid of the forward strand and see that all of those other ones disappeared. And also you'll notice all of the red ones have disappeared from the RNA-seq reads. That is because in this case, with apologies to our attendants who probably have issues with color blindness, red from blue sometimes it's easy to differentiate, but sometimes it's not. So you can be sure of what you're using for your gene model by removing the other strand. In this case, these are all the RNA-seq reads that are aligned to this particular model and that are the directionality is reverse strand. You can also see that by clicking on the details for that particular read, and it'll show you here. See that little minus sign over here? It shows you the position, and that tells you those are reverse strand. If we display the plus strand again, then all the red comes back. And if you click on any of them, you can see those are positive reads, or reads aligned to the positive strand. Not all RNA-seq data has directionality. This one happens to have it, so we can differentiate that way. Not that the data aren't produced with it, but it's just simply not included in the file. All right, how are we doing?
Okay, I just wanted to say that I've reassigned everyone that was using the Victorian machine number 5. So please go back to those user account spreadsheets. Depending on, you may actually have a machine in Queensland, or you may have one at Victoria. But just please have a look at those, and everyone, all of the 9 remaining machines are up and running. So, how are all of these sites going? Do we need a short little break to sort of regroup? Or shall we power on? Maybe we should just have a couple of minutes.
Yeah.
You could probably do with a short break. And then we could, is that good? And then we start the next section?
Yep. Sounds good. I'm going to start my timer here, yeah.
Okay, we'll just start in really just a couple of minutes.
Brilliant.
So there's a question from SBI. I guess we're going to start the exercises next. If people are late getting back, will they miss anything?
I muted myself and then I forgot. So before we start the exercises, we're actually going to do a little bit run around through Apollo functionality.
Yeah.
If they're late getting back, they will miss something.
So let's do 2 or 3 minutes from now. We'll start again at...
Yeah, no worries. The good thing is, so the majority, what they will be missing at the beginning are the things that I was just showing you, how to create a new gene, how to drag, or how to do things like that. So I'm just going to go through all of those little things that I was just blabbering about in a more organized way. And also you have slides that you can keep with all of that information. Oh, yay for Big 5.
It's back up. Well, I've reassigned everyone anyway, and I've moved all of your names around on the spreadsheet, so you can choose to use it if you want to. So in the meantime, are there any questions from any of the facilitators at any of the nodes about anything in particular? I guess people have generally been able to log in. There's a question from Mike Charleston in Tasmania. Is Apollo free to use?
Yes. Correct. So this is open source software. So not only can you use it, you can also modify things to customize certain aspects of it. And it's all available on, all the code is available on GitHub. You can download it from GitHub as well, so you can set it up for yourself. And this is all supported by funding from the NIH and the Office of Science of the Office of the Director of the National Laboratory System at Lawrence Berkeley Lab, as well as the Department of Energy and the University of California, Berkeley.
And from this end, so we're using a Nectar disk image that's running this training instances, but that I think we should probably investigate getting an Apollo up in a disk image that's available through Nectar as well. There is an Apollo Lite, I believe, that's available through the Genomics Virtual Lab as well.
Oh, on Nectar.
Yeah. So Mike, absolutely the person to talk to is Nick Rhodes, who's actually here. He's one of the trainers in the QSIF site, and he's been setting up the instance on Nectar, so he knows what to do.
Thanks, Jeff. It's just a question of how much time it takes and how much interest there would be to see if it's worthwhile doing. But thanks for that. That's brilliant.
Yeah. And if these instances that are being facilitated for this particular exercise are not up for very long, there's a demonstration instance that's always up in genomearchitect.org that people can use for the exact same data while you guys get, set up with having your own data sets. Nick also has all of these data that we're using for the demonstrations, so that if the time comes when you want to train a group of people and you want to use the same exercises, they can actually use all of these data. Again, all of the exercises are publicly available on GitHub, as well as now some of the solutions via Jeff. So it's all reproducible for you.
Yes, and there's also a comment from SBI saying they have some service set up in UNSW, but I need some help from Nathan to get it running smoothly. So
Oh, brilliant. Yeah.
after this, I will be distributing Nathan's contact details and some other things to everyone that's attended. So
Yeah.
it's great.
He's very responsive. So yeah, and he'll be able to get you to walk you through it. There's good documentation about this too, but sometimes you just need a little help. Yeah.
Okay, so it's coming up to 11:30, which I guess is halfway through. So maybe we'll start again.
Yeah.
I should also just point out to everyone that this is being recorded, so and we can distribute the link to everyone if you wanted to catch up on something that you missed. So over to you, Moni.
All right, so let's get going with some editing functionality so that we can look at our exercises as we are now an hour and a half to go. So as I was telling you before, we were kind of like playing with it and becoming familiar with the window. So this is what the next few slides are going to be about, simply you becoming familiar with the window again. So this is how you start a new gene model. As I was saying to you before, you can either create a new gene model by doing a right click on the actual on the gene model that has been predicted on the evidence and creating one of these 9 different sessions, or you can create a new gene model by picking it up and dragging it into the user created annotation area. Super simple. Then I explained to you a little bit of information about the sequence track, so that actual piece of the assembled genome that we are calling the reference sequence. You can zoom to base level to reveal that sequence track, and you can also color that by the coding sequence from the view menu. As I showed you before, just drop down and click on that color by CDS frame. That should be able to help you with coloring it. You can also remove the reverse strand, as I showed you. You can also toggle the protein translation if you don't want to look at that. And now let's talk a little bit about the basic functionality that takes place when we're curating different gene models. So I've divided this between curating simple cases and curating complex cases, but really you're going to find out very soon they're all very simple. Hopefully you'll get into the swing of things and then become addicted to it, as so many have. So a simple case, I define it as a gene model that's either correct or nearly correct, and there's good experimental evidence in support of this gene model, and this evidence mostly completely agrees with that prediction. And any evidence that extends beyond the predicted gene model is assumed to be UTRs, basically. So this is a simple modification. I wanted to show you first. Oh, I think that I hope that you can see at the very top. On the right, I have the entire drop-down menu for all of the different functionalities. So you have the ability to retrieve the sequence. This you're going to do time and again, so you'll get bored and you will know this very well, get sequence. You can also click on the annotation, right-click on the annotation to get the GFF3 coordinates. These will become important if you're sharing this information with your collaborators to find a specific location, or later on when you're trying, if you're an administrator and you're trying to merge the annotations that were manually curated with the predicted ones. Zoom and come back out of base level like I showed you. You can use the function to edit information to populate all of the fields on the information editor box. I'll talk about that box in a little while. Of course, you can delete or merge or split gene models. You can duplicate a gene model. You can insert an intro in the middle of your gene model, or you can switch a specific
genomic feature to the opposite strand. You can use the third box of information, the set translation start or set translation end or longest or all of these, to actually tell Apollo to determine what the start and ending points of your proteins are, if they are being automatically calculated. But every time you conduct an edit, Apollo recalculates the open reading frame. So you can actually set the start and end of translation to avoid that. You can also use this fourth panel basically to dictate to the software to pick up a little bit of information from the, say, from the RNA-seq reads or from a gene model underneath, and ask Apollo to imitate the boundaries of that piece of experimental evidence in order to modify the boundaries of your gene of interest by setting the 5' end or the 3' end or both ends of your gene of interest, of that particular exon of your gene of interest, according to the ones that are part of the experimental evidence. You can, this is fantastic, and you're going to love doing this later on, because you can set up, you can move basically the boundaries of the exon according to the next available canonical splice site. Remember when I was telling you that if I am an intron, on my 5' side, I have a GT, and on my 3' side, I have an AG. And so that GT site is called the donor site, and that AG site is called the acceptor site. So take that into account. When I'm talking about splice donor and acceptor, I'm always talking about from the perspective of that intron. And again, at the bottom of the display in this menu, you have a panel that allows you to do reverse undo and redo functions, much like you do on your word processing software or in any other software where you'd have those functions. You have a history, and I'll show you the stack, and I'll show you how the stack of history can be modified. So, for instance, say that you're going to add an exon. So here's how it looks like. You can either pick up the exon and drag its ghost up to the user-created annotations area, and then drag it left or right to reach the gene model where you want to add it to. And you'll see that there's this green light, or this green outline, sorry, that boxes in the gene model that's receiving the particular piece of additional information that you're trying to drag in. Once it turns green, then you let go, and now you've added an exon. This is another example of how to add an exon that's supported by the experimental data. So in this particular gene model, all of these RNA-seq reads are telling us that there should be an exon included in this area. So that's how I started. I pick up one of these reads, then I drag it. You can also add UTRs, and in order to extend UTRs, you can either go to the gene, to the portion of the boundary that you need to extend the UTR from, and once you hover over the end of one of the boundaries of the exon, there's going to be a black arrow that shows you that it is time to start moving the boundary of that exon. Else, you can do what I was just telling you about, which is to ask the software to either put an acceptor or a donor upstream or downstream from where the boundary currently stands. You can match the exon boundaries to the evidence, as I was telling you. So for instance, here we can see that all of this transcription evidence shows us that the boundary for that exon should be a few base pairs before we're upstream of that. And so you see that there's this orange circle with an exclamation point in the middle, which is actually showing us that there is a non-canonical splice site. So I can tell Apollo, all right, compared to this particular read, pick up my exon, right-click, and set that read as the 3' end for this particular exon. That means that this is a gene that is being transcribed under reverse strand. Once you're done with all of the modifications, then you want to check for integrity of all those exons. And I'm going to repeat this again and again on the exercises, and what it means is, we'll look to see if there are matching edges. All of the exons are matching the evidence that has been provided. The edge matching function, I'll explain in a second, but you also want to verify the exon boundaries and making sure that all of them are canonical splice sites. Here's a little trick.
there's little 2 tips. You can use square brackets to scroll from an exon to the next in the user-created annotations area. And if there are more than 1 annotations in the same region, you can use curly brackets to scroll from one annotation to the next. I'll show you that in a sec. This is what I was talking about by edge matching. Say that this gene model over here is in the user-created annotation area. If I selected anything, any genomic feature that overlaps that same region, will have highlighted borders wherever the boundaries of the elements of that genomic feature on the top match the ones on the bottom. That's why it's called edge matching. So wherever those edges coincide, it's going to light up red. This orange circle with the exclamation point in the middle is what we will recognize as a warning for non-canonical splice sites. And then this is another example of editing functionality. You can move these particular boundaries from where they currently stand on the non-canonical splice site to match these reads over here on the bottom by either matching them to them, so do the same set as 3' or 5' in, but you can also just tell Apollo, all right, figure out what is the nearest downstream splice acceptor and move the boundary to that position. So if you do that in this region over here where the warning is, then notice that it has now moved a little bit before. Notice where these reads are, and this yellow portion of those reads are. That's where the predicted gene model was, and now it's right above them. You can also figure out a way to set up a start and stop sites if they are not automatically set, or if you want to let them stay still while you conduct the other annotations. Apollo will usually recalculate the open reading frame. Sorry, something got into my eye. It looked like too blinky. Okay, so now let's talk about more complex cases, which are simply ways to say, well, in addition to editing a gene model by itself, you can actually bring 2 gene models together. You can merge them, and you can do that by either doing this little right-click merge function. Basically, you click on one of the gene models, the left one in this case, then you push the shift key down and click on the exon from the other gene model that you want to bring together. Then you right-click and pull up the merge function. You can also do that by dragging that exon and making the little green addition that I was just showing you before. So you would have to drag the entire gene model. You can also split an annotation if there's evidence and support of actually this being 2 different annotations. And then you can also annotate frame shifts and correct single base errors, as I was telling you before, because there are some, of course, inherent errors in the sequencing process. So now remember this. This is very important. We're looking at the gene at the assembly over here, right? This is sequences are all of the assembly. And this is a frozen view of the assembly. So even though you're annotating the assembly, you're not actually modifying it. So the assembly remains frozen, and what you're doing is anchoring metadata to that particular portion of the assembly so that the editing engine now understands, there's an insertion here, or there's a deletion here, or I need to substitute these bases with these other ones. So there are several menus that pop up, and you can say something like, you should substitute the next 7 nucleotides to the right with these base pairs, or you should delete the next 15 nucleotides to the right, or you should add these many nucleotides to the right. So it's always done to the right of the position where you're doing this, okay? And you'd always do it, you can do it on the strand, on the positive strand or the left strand, but it's always done, I guess I should say, to the 3' end of the direction of transcription. And if you add, for instance, in that position over here, if you add a letter, an amino acid nucleotide G, then that's what that's going to look like after you've completed that. You can also correct cysteine containing proteins, that is, proteins in which the stop signal needs to be translated into something else and needs to be skipped. So you can simply tell Apollo, all right, set a read-through stop codon when you find this particular coordinate. And what it will interpret is going to be, all right, so I'm going to read a uracil instead of a stop. And the protein will continue until the next stop codon comes in. I know that in the past 4 minutes, I just flew through a lot of information, but not to worry. It's all written down in here, and you can go back and look at this. It's exactly what I just said, letter by letter, so that it has a little bit more time to process. We can also add information to our gene models, and you're going to do this in your exercise. You will use the information editor by going to this portion of the drop-down menu called the edit the information. So this editor contains lots of different things. There's a side for gene and a side for transcript if we are actually annotating a gene model. If we're annotating something that doesn't have an mRNA, then of course there's only going to be one column. I will focus on annotating a gene model in this case. We have also a field to include information about cross-references to databases, either your own or somebody else's public database that contains information about this particular gene model that you're annotating. All of that information can be entered in a binary form. So you just have you enter the name of the database and the name of the accession. It's not actually talking to that database, but it's keeping this information for you and your administrator. You can add a PubMed ID. So in NCBI, there is this collection of publications from the National Library of Health and the National Institutes of Health Medical Library. And all of these references are called PubMeds, and they have identifiers that are unique to every single publication within that collection. So you can enter the name, the number of that particular article of interest that has information in support of the annotation that you're doing, say in support of the assignment to a gene ontology term, or in support of the length, or in support of the possible function, anything that you've added about it. If you used a piece of publication to enter that information, then you want to capture that so that it's also included with the annotation. It is connected to the PubMed database, and so when you enter an ID, the Apollo will send you a little dialog box that says, are you sure that this is the one publication you want to anchor this information to? And so you want to say yes. And then you also have the ability to enter gene ontology terms, and you're going to have to do that for your exercise. You can type in the gene ontology number, or you can type the term itself, the name of the term itself. It is connected to the Golar database from the gene ontology, so it'll actually autocomplete. It takes a second to think about it and talk to Golar, so give it a sec and be patient. And then you can just click on it once you're done. And then you can also enter comments ad lib. You can, as an administrator, this would be restricted, or you can just enter free text. Lastly, depending on how it is being worked on your institute, you have a little status box that you can fill information into. This is only if your team has customized it, but is not included on the exercises. As I said before, there is this possibility to change, undo, and redo actions, and there's an entire stack of history that takes into consideration these editing functions. So just remember, every time you use this undo and redo functions, it's just as if you were working with your text editor. That means you can undo, but if you change anything when you're undoing, then the stack changes and you cannot revert to something that you had already done, correct? So you can undo or redo as long as you don't change any further editing, much like you can type the word, undo the typing, or redo the typing. But if you change something in that process, then the stack redraws itself. Okay, this is the checklist. I call it the checklist for completing the annotation. It's kind of like, what are the things that I have to think about? Like, follow this checklist. Remember to comment everything you do. Add a comment to inform the community if you didn't manage to finish the gene model, for example, if you're participating in a group like that. But these are the things that I think about when I'm done with my annotation. Are there start and stop sites for this particular gene model? Are all of the splice sites canonical or not? Do I have any UTRs? Do I not? Are there any experimental data that would support the presence of a UTR or not?
are there any gaps in this particular region? And you'll see gaps as question marks in the sequence. Obviously, there won't be any amino acids translated from question marks, so you'll be able to see that. Did I have to merge or split two genes or a gene into two? Did I have to annotate any frame shifts? And did I have to do any other type of editing? And lastly, add a name, add a symbol, add a description, information to connecting to other databases. Do I have every single piece of information about this gene model that I could possibly collect? And if so, then we're probably done with each of our exercises. So this is the data that you're going to have with your exercises. The genome of the European honeybee was sequenced and published in 2006, and since then it has accrued an enormous amount of information. So these are some details on the data that you're going to use. You are going to primarily focus on this official GeneSet version 3.2. In the exercises, I ask you to not reveal this one track because it's a little outdated and you don't need to confuse yourself with it. There's also several other GeneSets you're going to use from the protein coding sequence, from the transcript sequence alignments. You're going to use where are they? The forwarder RNA-seq reads, the nurse RNA-seq reads. Those are going to be the two that I keep going back to as you walk through the exercises. And there's a lot of other alignments that can be used to visualize things, protein homologs from different species. These are just to basically support whether or not a gene model is present in a closely related species or not. All right, so I'm going to give you an example. Again, you can use, I'm skipping through that because there's one applied directly to you, the stuff that I had in the slides. But here, I'm going to use one of the examples that I gave you in the exercises and walk you through it. If you want to, you can actually go to your sandbox and follow along by doing this yourself. It's going to take a little bit for the BLAT search to happen, but you can start looking at the region if you want to. So, so let's go through this. There's a comment on the chat.
I just put in links to the
brilliant.
exercises and the sequences.
Cool. So let's think about this one. We're getting to the top of the hour again, so I'm hoping that I will not steal any more time for you to do the actual exercise. So let's walk through this exercise together. This is exercise number 5. And I'm going to go slowly through it so that you know more or less what the feeling is going to be once you have to come to your exercises. As you will see in the spreadsheet, you may be directed to starting from exercise 1 or start from exercise 7, and then they become a little more complex as you walk through the middle of the exercises. So this is exercise number 5. It's still not very complex. So we know of ceramidase as an enzyme which cleaves fatty acids from ceramide, producing this sphingosine, which is in turn phosphorylated by a sphingosine kinase to form S1P. And so the ceramide, the SPH, and the S1P are bioactive lipids, and they mediate several different processes such as differentiation, adhesion, and migration. So we know, because I'm telling you, that the honeybee, the ortholog of ceramidase on the honeybee is actually fragmented. And there may be 2 or more parts to this gene model, and so we are being tasked with fixing this gene model. So the first thing I tell you to go and do on these exercises is to interrogate the genome using the BLAT function. So what I do is I've provided you with a set of sequences, and so this will be a good place to remind you, even though in the body of the text of the exercises there are also sequences pasted, I ask you to use the little text file because browsers are very picky and very particular. For instance, if you open this text file, the instructions in your browser, and then you copy the sequence, and then you go and try to use an Apollo, and Apollo is in Firefox, then it's just not going to read those sequences. So just to avoid those problems, I've provided you with a file with sequences that you'll open in a text editor, and then you'll copy the sequence from there. Bear in mind that you do not copy the definition line. So that top line over here,
this, you do not need to copy only the actual sequence. In this case, these are nucleotides, so we're going to interrogate Apollo using the nucleotide version of BLAT. You pull up the BLAT window by going to the Tools menu and click on Search Sequence. This window will pop up. You'll paste the information here. You'll tell Apollo that you want to use the nucleotide BLAT, and most importantly, remember me every time you do this because you will forget, or try to, is you search all genomic sequences. Okay, so you click there and then you do search. And this is going to spin a little wheel for a few minutes, and depending on how many people are doing the search, it'll take a little longer, but after a minute or two, you'll get a list of results. This is your typical list of high-scoring segment pairs, and you're actually able to navigate by looking at these little lines. So you can either hover or click on them. As you click on them, you will navigate through them, but the list of results is not going to disappear. So what are you looking for here? What you're looking for is those HSPs that have the smallest score on significance, right? The smallest statistic, the smallest probability that these things happen by chance. And so you want to see that significance value as tiny as possible. There are several places here in different groups, 8.6 on group 14, on group 51. Don't go for the ungrouped ones. Usually that's a portion of DNA that was just not mounted into any scaffold, so we called it ungrouped. Sorry, I'm losing my carpet here on the ground. And so we're going to click on one of these, and in this case we're just going to go and click on the first one. It'll make Apollo navigate to that region. Notice now that it's over here, we've navigated to it, and then you can click close to get rid of this window. The next slide shows us what other teams have done. Instead of using BLAT directly in Apollo, they've used BLAST, for instance. You can actually generate a BLAST search, and then you can generate the link to the JROUSE in Apollo so that you can visualize that region, and you can visualize the HSPs as a track. So that's some customization that another team has done. You too can do that, but we're not going to go any deeper into how that works. We're simply letting you know that there are possibilities in case your team wants to get a little bit more involved with other search engines for querying the genome. So I found this region. Remember, this is that, I'll go back very quickly. This yellow highlight for the around that exon, 1, 2, 3, 4, that's that yellow highlight. So I pick up this gene model by double clicking on it, drag it up into the user-created annotation area, and drop it to create a new gene model in the user-created annotation area. Why did I choose that one? Because that's the one with the highest scoring HSP that I could find. So that sounded like a good place for me to start. Does that make sense? Okay, I didn't hear no. So we drag and drop that first gene model. And I'm hoping that you guys are actually logged in and trying to do this if you want to, because you can also just go to group 8.6 and find GB40335, and you can do this exact same thing. So you can start working on ceramidase if you wanted to. So once you get this new annotation, let's look at the transcriptomic data that is aligned to this region. I've cheated a little bit here, so I've zoomed out to display the Apollo window and show you this is the gene model that we dragged in. You see it up here? And there are a few other gene models. There's another one here and another one all the way to the left. Now look at these aligned reads. These are also RNA-seq reads. They're not red and blue because they didn't have directionality data at the time. So they're just displayed in this way, but this is the same way to display them. Notice that there are darker portions of those reads and lighter portions of those reads. By looking at this and at these reads, I can see that it looks like, so this is the gene model that I chose, right? That's the one exon that I had seen at the beginning. This is where that gene model kind of ends. There's an additional dodgy, maybe not supported exon over here at the end. But it looks like these reads from this exon, the last one that has sufficient support, look at them. They go all the way across 20,000 base pairs. And they end over here underneath another exon from a different gene model. So this seems to say that there's a large intron and more exons, way the heck 3' of this gene model that I predicted. So if I zoom into here, that's what I wanted to show you. See all of these transcription reads in support in support of the exon, nothing in support of this dodgy little exon over here, and then all the way to the other end. So I'm going to pick up that gene model that was all the way on the other end, and I'm going to drop it in the user-created annotations area as well. So now I've zoomed in to look at the other one, the tiny little one that had those reads 20,000 base pairs down. And I'm going to merge both of those transcripts because I think that that gene model on the right is actually continued on the left. It's just that because the algorithms that predict the genes are so greedy, they actually decided that these were two different gene models, but we're going to show it better. We're going to tell it who's boss. So we select an exon from each of the gene models, holding down the shift key, right-click to merge them, and now those two gene models are going to show up together. Yeah, I know this sounds like a little ethereal still, but if you're actually looking at this in the gene in the screen, then you'll be able to do it. If you're then you're going to see these two orange circles with an exclamation point in the middle, which meant non-canonical splice sites, right? So now we need to fix those. So the next thing that I need to do is to go and figure out how does the transcription data support or not the existence of these exons or the boundaries that they currently have. So let's zoom into this little one first. So what was the last exon of the first gene model to the right at about 1,400,000? If we zoom in there, we can see there is basically no evidence of transcription in support of that exon. This gives me confidence to say this is a dodgy exon indeed, and I can get rid of it. There's no other evidence in support of it. Sometimes they have like some transcription, some it doesn't. So we get rid of that one. And now let's go to the other end. This is the beginning of the second gene model. So this particular exon has this long boundary all the way out here into the area that has no evidence of transcription. The reason why this happens usually is because the algorithm that's trying to predict where the gene is has to find a methionine somewhere. So it invents these artificial tiny exons with, it could be just like 1 or 2 amino acids long, so that there's a methionine for that particular gene model. That's just an artifact of the prediction. So by looking at the transcription data, we can see that obviously an exon has been defined and the boundary should be moved from over here all the way to about here. So I can choose one of these reads and hold down the shift key to click also on the exon that I wish to modify, and then tell it, all right, set that one as the 5' end. And when I do that, then Apollo actually moves the boundary all the way to match all of these. Right? So that's what we were going to do. So this is group 8.6 where there's GV40335. I'm going to navigate there so that you can see it as I navigate. I can look at the group from here. Actually, I can actually look at the gene model. What was it for 0335? See that it's listed there. Oh, I've lost my, there it is. So I have a panel that tells me, which one are you looking for? I'm looking for the one on the official gene set. Go there. Oh, somebody's been working on it. Look. User 27 already started this example. That's brilliant. In the same space where I am. So what I can do is I can drop this gene model and then I can rename it so that we don't get confused. So this is going to be ceramidase, right? I'm changing, oh, actually, let's not change the gene. Let's just change who's working here. Is it CMD? I can't remember what the... And then this is just a second transcript. Look that it's already thinking that it's a different isoform because I'm dragging an additional isoform into the same region. But this will help us differentiate that I'm working with it. So if I zoom out very far away, I should be able to see that other gene model here. There it is. See? And look, somebody already deleted that gene, that exon. That was awesome. If somebody's looking at it, then they can follow along. See, there it is, ceramidase. So I would rubber band into here to delete this gene model. Sorry, this exon. And zoom back out to go look at that other boundary. And you can see how here, by shift down, holding down the shift key, I can then say, okay, set this as the 5' end for this exon. Why 5' end? Because this is a reverse transcribed gene. And there it is. Now we've imitated the other one. Okay.
There's a question from SBI. It says, our gene set seems to be missing the little gene. Can you give me the number again so I can check?
Our gene set seems to be missing the little gene.
I'll unmute you, SBI, and you can talk directly.
Oh, the second one, the GB40336? Maybe it's just too far away because the gene sets are the same. If you're both on group 8.6, the first gene that you found was this one. But remember that there was another region of similarity that you may have ended up in by simply virtue of going to the other good. So if you went to group unlinked 14 or unlinked 51, then you didn't find that same region, right? So what we were looking for, where we originally hit, was this region over here. We can highlight it. Where was that first exon? I think it was, oh, this one, I think it was the first one. Is the first one that we saw. Ah, I see. I know. It gets tricky if you don't see the arrows. They said that they were confused because they couldn't see whether they were pointing in the same direction. Yeah, remember that I cheated because I asked it to not show me reads in the forward strand. In this case, you could have just say, don't show me any genes in the plus strand, and then you won't be confused. Okay, let's finish this because I want you to do an exercise. Once you're done, so we already did all of this and deleted, and once you're done fixing the boundaries, then what you do is you retrieve the resulting sequence with that get sequence command that I told you. So that means you just pull out this collection of sequences. You can have the peptides or the coding sequence itself, genome sequence, plus or minus other nucleotides. And then with that information, you go to the BLAST service over at the NCBI website, and I also gave you that information. And then you interrogate the NR, the non-redundant database. Two things that I said on the exercises for you to do, I'll give you more detail about this later, is adding, making sure that we don't include the European honeybee genes from the BLAST, from the NCBI database, just so that we're not redundantly hitting the same set, right? So we want to exclude that from our database. And these are the results that we get. So this is like the cooking show where I don't let you wait for the cooking time, but rather I show you what the results are going to be. So I retrieve that sequence, and then I can say already that there's some conserved domains that highlight the presence of the ceramidase domain in here. And these are the HSPs. You know, there are a few other bees in here, bumblebee, the giant honeybee, Apis dorsata. There's also the dwarf one. So all of those are good indications that we've rescued a good chunk of that gene, of the gene model that we were expecting to see. And the cover is 100%. That means it's covering, the query is covering the entire HSPs that it found within the database. So that's also good news. So I'm satisfied with my gene model, for instance, and in this case, I'm just going to say, let's add all of the information that I can about this gene model. So that's where I would go and enter information about the name, the symbol, description about the gene. I would also enter information on the publications that I found on the genes, you know, say that I had a publication or that I went and looked at publications related to this gene for other species, enter information about gene ontology terms describing those functions from places, from closely related genes and NCBI, or say that I had that information. This is where all of that would go. So specifically for I think I have an NCBI search over here that's already working. Yeah. So specifically for the exercises, I'm telling you to go look at the gene pages for the particular gene models that we're annotating on the NCBI pages for Drosophila. And there's instructions on how to do that. And the reason why you do this is because there's a lot of information that is not available for other species, like articles that have been associated with that particular gene, as you can see, or down at the bottom, a collection of, where are they? A collection of gene ontology terms that are associated with that particular gene as well, right? And so under the gene general information. So when I tell you, go look at the NCBI pages for the Drosophila, this is what I mean. And all of this is explained on the exercises. So let's not talk anymore about this, and let's get you to work on these exercises in the next few minutes. And so what I would like for you to do is to open that document that we gave you with information for how to work on these exercises, and just take into consideration a few of these recommendations that I'm giving you at the beginning. For instance, let's know that the purpose of reading through these exercises is not for me to tell you step by step what to do and how to do it every single time, but rather for you to understand a little bit of that mind space of what kind of information should I collect as a curator, as a scientist who's trying to curate a gene model in order to improve the quality of the prediction and benefit the greater project, right? So these are not step-by-step questions and answers, but rather a little bit of, I'm challenging a little bit your thought process to try to understand how to improve this gene model. The first one, as I said, and the seventh one are going to be a little bit easier to get yourself a little bit ease into it and get your feet wet on it. The rest of them will become more complex. 5 is not very complex, so it's not all of them difficult. The sequences are in this file that you have already downloaded. And yeah, how about you give it a try with exercise number 1?
So Moni, will people try exercise 1 or 7 now?
Or 7 according to, yeah, you can do exercise number 1 or 7, sorry, yeah. So here's number 7 as well, according to what your table says. So in the
table has the order of the exercises assigned to each user.
Yeah, exactly. So here on the one where I am, it says that I'm going to work on server 1 and I'm going to begin at exercise 1. But if I am Jarrett working at GCU Townsville, I'm going to work on server 1 for organism honey 1, but I'll begin at exercise 7. And the reason for the contrasting places to start is so that you don't both start working on the same place. And I'm going to delete my gene model over here.
I guess it's also worth mentioning that don't stress if you don't get all 7 exercises done today. This is, I guess this is a awareness raising activity and making people comfortable with the Apollo environment. So I guess, will you just quickly go over 1 and 7 in a bit, or will I guess?
Sure, we can. I would like to give them.
How would you?
I would like to give everybody the challenge of reading through the instructions and start with the exercise, and after a few moments we can work on them. But let's get everybody soaked and troubled about it for the first few minutes, and then we'll do the explaining.
Perfect. So should we say 10 minutes? We'll play it by ear.
Yeah, I think that's fine. 10 minutes for the first one. And as Jeff said, we won't get through all of them. It takes, some of them take a little bit more time. And it's not the goal of our meetup to get through all of them. That's why there's a demo instance that you can visit later. Hopefully, you can do that. Maybe the instances that are currently up can be up for another 24 hours or so, and that can help you walk through them. I'm going to quietly start working on 7 if someone just kind of wants to follow along. I won't be talking, but I'll start looking through these instructions again and slowly going through the process. So I heard that there's a question regarding saving the history of an annotation. So there isn't a way to extract, to export the history now. As an administrator, you can actually collect what the different editings have been across all of the genome. And so you could programmatically query editings that have happened for a specific annotation, I guess. But this would have to happen programmatically by the administrator. The user does not have a way to save those. What you can do is record them by text, I suppose, if you really need to save all of that information. Does that answer your question, Brad? How would you record it? So as the editings take place, I'm going to get out of this area because somebody's going to end up here somewhere. As your editing takes place, then you can type information about what functions took place. So for instance, say that I am going to bring these three together. I don't have to, but if I have this gene model and I know that it needs to be joined to this gene model, for instance, then I would merge them. And on the resulting gene model, I would include information on the editor mentioning something along the lines of in the comments. So this gene model is the result of merging GB4030 and GB40341. 40340 and 40341. So this would be one thing to do. As far as keeping track of what editings are taking place, you would have to enter them, or your administrator could actually create canned comments that you would be able to just choose from a drop-down menu if they were included. So for instance, my only canned comment in this instance is testing, but you could add another one in there. Having fun trying to drag our found sequences into the annotation. I imagine that by having fun, you mean you're having difficulties?
Yes, it says, yes, we are having difficulties.
So I wonder, I don't know. So my only thing is sometimes the newest Mac track, the trackpad, the number 2 has like a third button, like a third level of pressure that can throw you for a loop, but it works for me, and regular mice do as well.
It's not that, it's that from our search results, we do the BLAT, and then we end up with a bunch of results, and then it's a question of, okay, how do we turn those into...
Oh, I see.
Yeah, so I don't know whether we missed it just went by too fast, or...
No, that's
struggling to do that.
Understandable. We can do this. We can do a BLAT search here so that I can show you that.
Cool.
This will take a few seconds as it's querying a pretty big chunk with BLAT. Did anybody order pizza? In the meantime, is there a way in the settings somewhere to consider the T2C change in the splice site turning GT to GC canonical? So that is not this place for these sites.
All right.
No, so there's no way to make the non-canonical splice sites that are canonical. But that's a fantastic feature request that I have been asking for years, and I left the project without it being realized, but it would be an awesome GitHub issue to file. So Mike?
Yeah.
You can see my window here with the search results, right?
Yeah.
So as you hover through the results, they turn blue. But when you click to the results, it actually navigates to that HSP.
Yeah, that's
so
all good.
Here, huh?
That's all good so far, yep.
Yeah, and so here we've navigated to this particular gene model, 8.6, blah, blah, blah. And so that's the first one. It's found this one called 40348.
Yeah.
Mm-hmm.
Still all good. So how do we make that into an annotation?
Okay, so I just leave the search.
Right.
Right, now I just leave the search. And I'm gonna get rid of these RNA-seq reads for now, because I don't want them to be in the way. And now this highlight is kind of annoying to me, that bright yellow. So you can just click on the highlighter X. See, that was my third button here. Now, another thing that I like to do is to change the scheme to make it dark so that I can see a little better because I like terminal windows. So to create a new annotation, you double click on it. If you don't think that you caught the entire thing, you can zoom out a little bit to see where you caught it. So you can double click on the gene, or you can also click on the intron.
Yeah, we can drag that up.
You can right click on it and say, create a new annotation. So that's one way to do it. And if I want a gene, now there's one in the top. Did you see it?
Yeah.
The other way is you can click on the intron, and now you drag that ghost up. And you see how it moves as I move?
It's just a question of going from that search results window out to seeing the highlighted region. And that's where we were struggling.
Oh, sorry. Yeah, all you have to do is to close the search box.
I think that's all right. Yeah, cool. We'll give that a go. Thank you.
Sure.
There's another comment about the history of edits, which I believe there is.
Oh, so the history of every edit that you've done is available here. That's what I was telling you about the undo and redo. Every edit is recorded. You cannot export this. There's not a format to export them. For the admin, there's actually a report that you can generate on changes. So this report would actually tell me everything that is going on for every gene model. But the user does not have access to that. But it's, so do you mean to access the trace or history of the edits in any other way other than everything that I'm doing to it through the history in this panel?
Unknown. The question was from Adelaide.
Mm-hmm. I think you can circle.
Yeah, I mean, feel free to type the answer in the box.
Okay.
Yeah.
So I think I used, for number 6, that's number 7, sorry, that's what I was doing here. And that was my first gene model. So let's see what else do my instructions say. Left to right to left. Retrieve their sequence. Can you repeat the question? The question that was, it was posted on the gene model, sorry, on the chat. It says there's a feature request based on the comments. It would be really valuable to the users to be able to access the trace or history of the edits. What I asked in response was, do you mean something in addition to the history panel that already exists? That was, this history panel. At the moment, it only has one because I've only dragged it, but every edit will be recorded there. So now it wants me to get the sequence. And interrogate. I always change this name just to have a smaller thing that I can recognize. What was the name of this? Gene APC. Correct. The administrators also have access to the history in the entire server. Here's something that I was saying earlier, that I want to exclude the honeybee. So you can just type Apis mellifera. and ask to exclude. And if you want to further restrict the search, you can just use this taxon in this case because we're working with honeybee, and then just leave it that way. And let's see what it says. It's a lot of hurry up and wait. Armadillo repeat. I don't even remember that. Let's see what this is. So right here is the collection of conserved domains. APC repeat. In the region of that nematous polypsis proteins, that's what I was looking for. I suppose. Is it complete or not? If I understood it correctly, the feature request was about the possibility of exposing the entire history of edits to the user. I guess you want something more specific than what currently is in the history, which would mean including things such as additions to the information editor, because everything else is recorded. So I guess perhaps you think there's a few things that are not recorded and should be kept. That would be fun. Why don't you can go to this site. So you have to have a GitHub account, but of course those are free, and you can enter a new issue here. Once you try to create a new issue, there's going to be a set of instructions that tell you what exactly are you trying to do. But that would be fun if you want some more specificity on the things that are kept on the history track, and so the Apollo team will give it a sense of how quickly should this happen, if it should be happening, or what modifications can be done to update what's currently available. Ah, that's interesting. Ah. Uh-oh, somebody raised my gene model. How dare you?
And this is an interesting topic that I guess when you're actually using Apollo for collaborative work,
Yeah.
there needs to be some ground rules about, within a team about how it's used. Do you have those, or have you used those
Correct.
for the
Yes.
honeybee project, etc.?
Yeah, when I am actually addressing a group that it's already engaged into the annotation process, I have a whole set of ground rules that can be established by organization. And so different groups play with it differently. You can establish limits on the permissions that people have when they look at somebody else's gene model. They can see it, but not edit it, or sorry, they can see it, but they cannot erase it. And so that could be the case. So here, someone just saw the annotation and said, "Nope, I'm going to work here," so they got rid of it. But it's So what we usually try to stress is that since we're going to be working in a collaborative environment, then the best way is to come to an agreement. If you have, to come to the same place and the same space, if you have differences of opinions on how these two gene models or this one gene model should be addressed, and also divide the load. So in here, because we have a limited amount of space, and we are trying to do the welcome everybody into the same space, then we're bumping into each other here and then. But in this case, what I should have done is to label it so that when they saw it, they just saw, this is Moni's gene model. Even though when you hover over it, you'll actually see that it had my name as the owner there.
Yeah,
in
great.
that little one.
And I also had another question. So when in a collaborative project, you're satisfied that you've come up with a really nice annotation. So you spent a lot of time doing this manual curation. Could you just maybe talk a little bit about getting it out of Apollo and into a model organism database or some other type of resource like that?
So what traditionally happens is that when you're working on this project with a group of people, once you determine that all of the annotations need to stop, so you make a freeze of whatever has happened. And 1 or 2 rounds of quality control will have to take place. Different people approach that differently. Some groups just go one by one and check. Others have scripts that run through all of the the manual annotations and say some things need to be flagged and need to be fixed, and you approach whoever the person that did the annotation is in order to fix them. Once there comes a time when the time comes that all of the annotations are exactly the way you want them and the manual annotation should stop, then you restrict access to everyone, and the administrator downloads all of the manual annotations, and the process of merging begins, which usually, depending on, so some groups work on this differently. Some groups say, okay, you only have to annotate things that should be added to the official gene set or that should be removed from the official gene set. And so by the time, when it's time to merge them, the scripts will recognize things that are overlapping and say, okay, this manual annotation supersedes the predicted annotation that was in that region. you should indicate that if there are more than one isoform, all isoforms should be annotated, and those kind of rules take place in order to do the whole merging process. But eventually, you will have a finalized set that includes the manual predictions as well as the automated predictions as well as the manual annotations, and that will be your finalized official gene set. The communities approach it differently. I'm actually really pushing really hard. There's one community who has scripts in a set of rules to work through this so that it can be public and sent out to everybody to emulate. Others simply do not go through the entire process of merging everything automatically, but every so often, after they've collected, say, 6 months of manual annotations, then they stop everything, freeze everything, make Apollo unavailable to everyone, and then they manually populate this information into the database. That's a lot more painstaking, of course, but there are some groups that choose to do this in a manual way. So I'll be working with students who definitely want to stop them from deleting what other people have done. So these restrictions on who can do what when have to happen at the administrator level, and the whole thing about preventing somebody from erasing your annotation, it's a newer feature, so that would have to be done, I think, on the back end. This is all programmatic still. I don't even remember whether that exists on the administrator panel. It didn't when I left the project in August. I don't see anything on permissions other than reports. Yeah, so this has to be done programmatically. Good question for Nathan. If people want to work with their own data, how do they and can they set up their own Apollo server? Should we follow up with Nick? If you're going to set up a server within the EMBL-ABR instances or their servers, then definitely you need to talk to Jeff and Nick about this. For anyone who wants to use Apollo on their own, there's a little bit of sysadmin involved. You have to be able to be knowledgeable and install, for instance, things like Apache. There are a few dependencies. You have to have Apache. You have to create a Tomcats instance. You have to be able to run all of this from the terminal window. You have to be able to set up and move the data. So it's not trivial for someone who's not accustomed to doing a little bit of sysadmin. It's not a desktop application that you can just download. However, once you set it up, you can run it locally in your own machine. You can use your laptop as your Apollo server, but that would be a very private instance, and there would be no sharing with anyone unless you open your laptop to the internet for collaboration. So in the case of working with the EMBL-ABR nodes, then this would be something that takes place by talking with Jeff and Nick.
Yes, and I think that that's something that I'll definitely be suggesting that the EMBL-ABR looks at, is making that easy for people to actually spin up their own on a Nectar VM, for instance. I did have a question though about management of that VM, and how often are there releases of Apollo? And so, obviously if we set up a VM with today's release, there'll be more releases, and I guess I'm just interested in management.
Yeah. So up until I was managing the project, there were somewhat periodic releases. By that I mean every 6 months approximately we would have a new release, depending on some pressing needs. We would do something that was a little bit closer than that, but it's usually semi-annually released. And now, because I left, the last release was a bit of a flop. So, don't record this, delete it. But it was a little complicated. It was very complex, and there are things that are still being restored. And so a new release is coming up, and I think, so this past release was done in September, I think, and they're trying to get something out officially again in February, at the end, after the Plant and Animal Genome Conference. However, they're recommending that everyone just pulls from main on the GitHub issue because they have had some issues with the release. So unfortunately, if you're going to set up a images, you could probably roll back or just pull directly from main instead of using the latest stable release or the latest stable release. I would recommend just to pull from main because there were a number of issues that came up afterwards.
Sure.
But yeah, expect
Great.
about every 4 to 6 months.
Okay. And Nick's just actually sent me a message saying the image for this VM is actually public on Nectar, so anyone can go and get that, and it will have all the current services and all of these test data as well.
So someone just said, are you saying there are problems to that 0.7? Yes, there are. And all of the patches are available on the main branch on GitHub. I'm sorry to be the bearer of bad news, but it is. Yes, I'm so sorry. I started seeing all the stuff afterwards. I think there's a question for you there from Sonika.
Yes, so I've got some slides to go over at the end. These particular VMs, they're quite large, so resourcing them for this many people is a little bit challenging. I guess it's worth noting that Apollo has a demo server, and I'll put that, actually put the, and it has the same data in it as well. So anyone can go and play on that particular server whenever they like.
Yeah, so this is over on Genome Architect. There's an instance that you can visit with the fair warning that this is not an instance for production, which means it can come down and data can be erased at any time without necessarily announcing it in advance. It doesn't happen very often. It happens once every several months. But just in case you're unlucky and that happens the day you're working on it, just be aware.
Certainly, I mean, as far as the QSIF resourced VMs, I think we can probably just keep them up for a day or two. Someone at Melbourne Bioinformatics would have to comment about those ones. But as I said, the machine behind it is quite large, so it is a bit of a resource that we have to provide. So I put the link to the demo environment over in Berkeley into the chat box.
Ah, brilliant, yeah.
So we have about 14 minutes left. How would you like to manage that last, I guess, 10 or so minutes?
So we can, exercise number 1 is painstakingly solved in the videos that I sent for you to share. So it's a PowerPoint presentation, and it has some pictures and it has some video that you can look into in order to answer it. So why don't we try and work on 7?
Great.
Do you guys want to do 7 together? Just as an example. Number 2 is also explained. So let's just go through this here. I'll delete this that I was working on because we don't need it. So I'm just going to go through the information that I've given you on exercise number 7. It came to our attention that this ortholog of the the APC is fragmented into possibly 4 or more genes in the current gene set. In Drosophila, this gene is involved in beta-catenin binding and microtubule binding. APC is a negative regulator that controls beta-catenin concentrations and interacts with ecadherin, which are involved in seal adhesion. And APC is classified as a tumor suppressor gene in humans. Mutations in the APC gene may result in colorectal cancer. So I've given you a fragment from the Florida carpenter ant, Camponotus floridanus, and the gene model is also available in NCBI. So we're trying to query the apis mellifera genome in order to find it, and to do that we're using the BLAT search option. So we go to our little file of sequences and find number 7, which is this one, to query the genome with it. So we do this search my sequence is actually a protein sequence, so we would have to change the settings to flat protein. And then I've cheated because I'm already in the scaffold that we're looking for, but if I weren't, then we would be clicking on search all genomic sequences. We're not going to do that right now to save a second, but you would do that if you were not in the group that you were looking for. So so that's why this search was very fast, because I'm looking into one scaffold instead of all 5,000 of them. So I have a few hits here. And, uh-oh, can you hear me? Okay, sorry, I had an announcement of unstable internet over here. So I have these two hits within the same scaffold. And I had already pulled up some forwarder RNA-seq data. It seems that those are the top hits. And other than that, everything else is still pointing at the same gene model, even the ones that have high, 350. But these are very low significance values, right? So I'm just going through all of the results, trying to see what all is visible about these HSP. So I seem to see that there are two different gene models overlapping. Okay, so I'm happy with that one, the first one that I found, and I can close this window to let me see what's happening here. I've already pulled up some RNA-seq reads, and I'm going to get rid of that yellow highlight. I can get rid of that just clicking on the marker or removing it from the view menu, I believe. Yeah, you can clear the highlight. So the first thing I want to do is to drag this gene model that I found. Is thinking about it a lot because I have the RNA-seq reads in there. Let's get rid of them for a second. So I'm zooming out to look at some of these gene models to see, this was the one that had the highest HSPs. And I want to drag it up on top to get the sequence and figure out what do I have in my hands. So this was like 2,700 residues. As you can see over here, this is a pretty long protein, several exons. And I want to go look and figure out what this is like. And so I would go to the blast site and query in our database. I've already asked to exclude the APIS. Oopsie. The honeybee genome. I can just show you that. Then I click to exclude, and I've asked it to restrict the searches to Insecta. I can paste that over here. And I like to change this name, because it's too long. Just because I think I have named something similarly, so the job title will show up here. So then I just say, hit it. So the cooking show shows me the results over here for this particular search. And I have up here on the blast search, there are these, there's this information from the conserved domains database that I'd like to look into. You can look at them in here, although it's a little complicated to the code. So when you click on it, it actually sends you to the page that has this exact query, but it has all of the information regarding conserved domain database. So just to remind you, the data we're looking into is APC, this adenomatous polyposis coli. And this is the collection of identified conserved domains that we got from the sequence that we retrieved. So there's something called the armadillo beta-catenin-like repeat. okay, that sounds promising. Those are things that should be there. But there's also another one called the APC repeat. And so this is a short region found repeated in the mid-region of this particular set of proteins, the APCs. So promising, we found an APC domain in what we think is an APC protein. And that's something positive for us to see. So that tells us we're on the right track. These are, of course, alignments of the HSPs. These are not as informative as it is to actually look at the alignments. All that it says is like, okay, we have pretty good sequence similarity to some other proteins in there, but let's go look at them in detail. So down here, what you see next is a description of the HSPs with their species. There's several isoforms of this one protein in Apis cerana. There's also Apis florea, so these are all bees, Apis, all bees, bumblebees, solitary bees, the ginormous one, the giant bee, wasp, polistes. Yeah, we have a lot of different, ants, Adocephalides, leafcutter ant, fire ant. So those are the list of species that are that have high sequence similarity, with which my sequence has high sequence similarity. But here's the actual meat of this. We're looking at the alignments. And the first thing that jumps to my mind here is that this is my query. In my query, the similarity, the high sequence similarity begins about 50 amino acids into my query, and actually about 130 amino acids into the subject sequences. And this is true for the Apis cerana isoforms, again. So that means the beginning of my protein does not match exactly the beginning of the proteins in the database. Again, with Apis cerana, here's another species, same isoform, another isoform in the same species. This is an Apis florea, another bee, and the same is repeated again and again. That and what I'm seeing on these conserved domains tells me, well, maybe I'm missing a chunk of my protein at the beginning, right? And so by that consideration, what I should be looking for is whether there are any gene models predicted on the 5' end, upstream of the 5' end of my protein, and also in the same, being transcribed from the reverse strand. And one way to figure out whether there are going to be reasonable gene models that I can use is to look at transcription evidence. So I'm going to pull some RNA-seq reads. And I need to zoom in, but I'm gonna do a little cheat that you guys can learn how to do. You cannot break this. So you can do this and mess it up and do it again. If you edit the configuration of the RNA-seq reads file, you can display more reads. So I'm just going to do this so that we have a little bit better zoom capabilities. So we need to zoom in a little bit. Hopefully we'll get some reads here. Here they come. You can do this. There they are. So looking at this, just by looking at this picture with the reads that are still loading, I can already see something. It seems that there are some reads, there they are, yeah. So remember that the lighter part are supposed to be connections between portions of the genome that are transcribed and were captured by the RNA-seq reads. So I can see that some reads that begin over here in this area, and they are actually under this predicted exon, are connected all the way out here to this exon, which is very well supported by all of the transcription here. You see that? So I would be inclined to try to merge these two gene models together. It's very likely that they belong together. And it's very likely that this exon, which is mostly in its majority not supported There are some residual reads, which are the effect of secondary like read-offs when the sequencing is taking place. So I would say that this exon here is probably dodgy, and I'm going to get rid of it. And then over here, I would know, oh, I don't know what to do with this exon. There is not much support for it. However, the coding sequence is recovered. Let's see what happens if I delete it. I still have a coding sequence. So let's see what the exercises are saying is now that we do now. Once you bring out the information, stop and observe the direction of transcription. Does it go right to left? That's just to figure out whether it's reverse or forward transcribed. Drag the annotation. We did that. Use the product to query the blast, the NCBI with a blast, and we did that. In the NCBI pages, we reviewed the conserved domains. We found the conserved domains. They were intact in the gene model that we looked at. But we thought that there were two models that needed to be brought together. So we merged those models together and rearranged the boundaries at the joins. And we're trying to figure out whether this has now rescued everything that we needed for this gene model. Right? So what we have done, there were how many amino acids before? Just a little bit over 2,200 and peak. Go back up. This is a tad longer of a protein now. And after merging them, we should try to figure out whether we've recovered that first part of the alignment that we were missing. Sorry that this is being so finicky. ECP or APC? Why do I keep forgetting?
We're also up to time, so
okay.
If we maybe just spend another minute or something, and then maybe we can think about wrapping up, would be great. Thanks.
Sure thing. At any rate, so what's going to happen here, likely we'll see that the alignments are now a lot closer, and that means that we've recovered most of our gene now. And after we realize that we have actually captured the gene completely, then what we want to do is to enter information, more information about it. I'm going to get rid of this reads because they're making the client hiccup. So as I was saying, you can enter information about it here. We had this very long name for the gene, and that we would enter here. And this would be the symbol. The same information would go here. However, this is the isoform, and we need to give the isoform some type of convention. The R comes from the conventions from the Flybase team, which generated this for everybody 2 decades ago, which is the first isoform should be called RNA form A. That's where that RA comes from. And then, or you can just call it 001 or something like that. And then all other information that you have captured about this would be entered here. Details on how to enter PubMed IDs and gene ontology terms are also available from the little exercises that I give you. I know that 1, 2, 3, 4, 5, 6, 7, 8 happens to be a PubMed ID. So I'll show you this as an example. It's a sociology paper, I think. So it's actually going to ask, oh yeah, Declaration on Population Development? Sure, that's the paper we want to enter. And then the gene ontology terms are entered in the same way. Where do you get this is from trying to figure out, from finding the gene, one of the genes that are very well curated from these HSPs, which is usually a honeybee gene if you're talking about arthropods, but it could be a human gene, a plant gene, a Rhabdopsis gene, depending on the species that you're using. So as I think I had mentioned before, I have, this is the information. You would go and look at the gene ontology, say that this is for example number 2, I think, or 1 for the small nuclear protein complex. So you would go to the actual page with the Go term, and you can get information from there, as for instance the accession number, or you could get from the references, get the PMID down here, and then add that information over here in the in your information editor.
Fantastic. We just have a couple of questions that have just entered, so maybe if we could just go over those. So Sonica
Sure. Can a Apollo user be restricted so as to not expose the editing features, that is, to behave more like a vanilla JBrowse? It's called JBrowse. So what you would do is to simply not give access to Apollo. You can make your genomes public. So when you can just simply give the URL to JBrowse. And actually, you can make it so that when people log out of Apollo, all they see is the JBrowse window. So they can be exposed, but the annotations are not visible, only the predicted material and the experimental data. So there's no way to expose annotations without editing. Well, there is way to expose annotations without editing power, but there's no way to expose annotations without logging credentials. Could you please again show how you edited the JSON file to include more RNA-seq reads in the track? I know, Sonica, this is going to get you into so much trouble, but I really like it because that's the only way to reveal reads sometimes. So once you see the label for that particular track, there's a config file here, and that's where all the JSON mumbo jumbo is. What I did in order to show more reads in a specific space is to change the feature scale. So you can change this feature scale property to 0.1. If you're really, really confident or you're working by yourself, you can also change it to 0.05. And so it'll show a very large number of reads instead. Does the JSON edit affect the server or just the browser? Just the browser. This is all on a per session basis. So you're not, that's why I was saying you cannot break it. All you're going to do is maybe make the client upset, but you're not changing anything in the background. This is specific to your session. Is there a way to easily find the annotated start codon for a gene? Oh gosh, no, there isn't. And it would be fantastic, but we don't have. I've been asking for this for solid 2 years to have the identifiers for the start and stops. You know how if any of you used Apollo, the desktop version, the Java desktop version from a million years ago, you remember that we actually have flags for starts and stops, but not in this one. Another one for that GitHub thing. Kearns is leaving the session. Sorry, bye. Will merging an upstream exon automatically extend the ORF, or do you need to find the upstream start codon too? Adding an upstream exon is simply going to make Apollo recalculate the longest ORF, but if the longest ORF does not include that particular exon because there's some modifier in between that exon and whatever was predicted before, it's not going to move it. So you have to find it as well. What in my experience I have encountered is that once you add that previous exon, you begin the modifications because now you're going to have boundaries that are non-canonical. So you begin to make those modifications, and then Apollo naturally recalculates what is the true longest open reading frame, because thankfully biology mostly always works. So it just naturally finds that longest ORF. You're welcome, Mike. I know you guys are leaving soon. Sorry that this is it's a lot of fun and the questions get exciting, and then we also had a bit of a hiccup on getting connected. But the good thing is there are at least 2 exercises for which there's slides and videos that are available to you. I have, as I said before, never intended these exercises to be a step-by-step answering, but I see the need for some cases when I'm not going to stick around to answer questions much longer. So I'll work on making sure that I do a step-by-step of the other ones so that you guys can benefit from that too.
And I just had a question before we wrap up. So there's a couple of, I guess, questions. So, and you're not at the Apollo project anymore. So you have fantastic online documentation and a guide, and we'll be sending out, you know, all of this in an email afterwards. But is there someone? How would someone ask, you know, for specific information? Is that possible, or?
It breaks my heart to tell you that there is not a money replacement. There is. So I managed the project for all these years, and I created all of this documentation. But upon departure, there is no replacement. And so, in all technical aspects, Nathan is your man, and he will be fantastic to assist you. In terms of the biological principles and the aspects of the actual annotation, there isn't anyone at Berkeley that can answer those. So your best bet is to reach out to groups that are conducting the annotations. And I am, of course, very happy that we were able to schedule this so that I could still do it. And so I'll be able, you know, I'll be glad to answer questions in the coming weeks for things that are related to the exercises. Unfortunately, in terms of further training for other teams and things like that, then that's going to be a little more difficult. I'm thinking I'm going to have to open a consulting firm of my own.
Yeah, absolutely. So, I mean, that sounds fantastic. And if you're happy for us to, I mean, maybe we can collect questions and send them to you. That's probably
and I can do something like a little video cast and you know, like have a powwow with your questions and just address them as you present them. And if I can, you know, I can do a screencast of things answering. I think that would probably work.
Fantastic. So I think we should wrap up. So hopefully you can see my slides now. I just, again, wanted to say a huge amount of thanks to Moni for joining us today. It's been really, really fantastic. Very, very happy. You've basically worked into the night to do this. You're obviously very passionate about Apollo, and we very much appreciate your time, so thank you. Also to Nathan and Nick for setting up these training environments, and QSIF and Melbourne Bioinformatics for resourcing those environments. And thanks to Simon for restarting the server. He's actually off work sick today, but he did that, so and that's back up. Really want to thank the facilitators at each node that has helped to put this together today. And of course, to all the participants. We've been really excited and sort of by the fact that it's been a very popular idea. And we've had, I think, about 90 registrants.
I think sorry, your screen's a little to the right. I'm not sure. I cannot see your entire set of slides, actually. So.
Right. Is that better?
No, I see a white board, but this might just be my settings.
I think what I'll do is just...
Yeah, there you go. That was whatever you just did.
Okay. We'll just leave them like this. So the training environments and accounts you've used, they will be certainly available for a couple of days, but I can't promise longer than that. As we noted, though, that you can go and play at the Apollo server in San Francisco, and that's available. And the last thing I really wanted to say was that today's training has been a trial for a new model of training delivery for Australia's bioscience community. So EMBL-ABR is really keen on seeing if we can try to do this by having experts, be they based somewhere in Australia or internationally, like Moni has. So we've been really, this has been a little bit of an experiment. I think it's gone very well. So thank you, gods of the internet. We would really, really like your feedback, because this has been a new model. So feedback from you on obviously the content, but also the concept and method of delivery of this. So we'll also send out this. I think that Christina has already sent this link out, but we shall be sending it out again. And that's it. So again, thanks everyone for joining us today, and thank you again, Moni. I think it's been fantastic, and we look forward to maybe, you know,
Well, we look forward to sending you questions.
Yes, of course. I'll be glad to do my best to get you guys answers as well.
Thanks, Moni. It was as good as having you in person over here.
Oh, thank you. I was brokenhearted that I couldn't go. It's still a thorn on my side. I wish I could have been there, but you know, the gods of visas didn't work this time.
Maybe next time.
Yeah.
Okay, well, thanks very much. We'll end the meeting now. All right? Thanks a lot.
Thank you.
Bye.
Bye.